Well, good morning, everybody, and welcome to the next in the series highlighting organic chemistry across Europe. And this morning, we are delighted to be highlighting organic chemistry in Sweden. So my name is Pat Gyrey. I'm from University College Dublin in Ireland, and I chair the division of the EuChemS Organic Chemistry. And it's my pleasure now to pass over to my colleague on that division, Professor Henrik Sunden, who will introduce and tell us lots of interesting details about the importance of organic chemistry in Sweden. Henrik, over to you.
Thank you very much, Pat. Thank you. I'm so happy to do this presentation. It's a true honor for me to stand here as the representative of the organic division in Sweden. I will tell you a little bit about our work with organic chemistry through the Chemical Society, and I will also introduce the speakers later on in the presentation. So, let's see here. Where do we find Sweden in Europe? Of course, Europe is in the northern part of Sweden, northern part of Europe, as you know. It's flanked by Norway, Finland, and Denmark. In Sweden, we have a chemical society that we call Svenska Kemistsamfundet. We have 12 sections, where the organic chemistry section is one of the larger ones. We have a newspaper that comes out 4 times per year. Giving out to the members. It's called Kemisk Tidskrift, and it was founded in 1889. The society is responsible for giving out several prizes and awards, and we do organize also several conferences each year, which brings me to the point of commercial. So I'm gonna say a few words about the upcoming third meeting of the Swedish Chemical Society. It's going to be held in Stockholm. Here you see the beautiful scenery that you can see in Stockholm. It's going to be in June 2025, and all naturally participants from all fields of chemistry are welcome here. And if you want to know more about this event, then you have a link to the web page here. And of course there are a few historical figures that you can mention when you talk about organic chemistry in Sweden. My pick is starting with Carl Wilhelm Scheele, who is the pharmacist who almost discovered oxygen. He was a couple of years late with that, but he was successful in discovering glycerol. He made major contributions to work with organic acid, so he discovered malic acid, mucic acid, citric acid, and oxalic acid. And he's also the first person to carry out and document the acid-catalyzed synthesis between ethanol and acetic acid, so the esterification reaction. And of course, when you talk about Swedish chemists, you must mention Jacob Berzelius. He's known as the father of chemistry in Sweden. And apart from discovering several elements of the periodic table, he's also known for coining several phrases that we use daily in our work as chemists, such as catalysis, polymer, isomer, allotrope, and protein. And he is also the first person that have used organic chemistry in writing. So the term organic chemistry comes from him. Here you can see it's a copy of the original book where he writes Organisk Kemi, which is the equivalent to organic chemistry. And this came out in his book on animal chemistry in 1809. And then, of course, I would like to mention these two guys, and partly because this is a fascinating picture. Here we have Nils Löfgren to the left and Bengt Lundqvist to the right. They're all set up in a laboratory with nice lab coats, lab goggles, and they're inspecting some kind of distillation apparatus. But Bengt Lundqvist is also smoking a pipe in the laboratory, and this is sort of tells quite a lot about what how we have developed with our security thinking in the lab. But these guys They developed a compound known as a drug called xylocaine, which is a local anesthetic. They sold that one to a company called Astra, which is of course the company AstraZeneca today. And this became a major selling drug and has sort of influenced the development quite a lot, and developed Astra into the pharmaceutical company that we see it as today. So if you want to go to Sweden and do organic chemistry as a postdoc or a PhD or on a sabbatical, for that you can do organic chemistry at several universities. Starting up north, we have Umeå University. And then further south, we have Uppsala University. Here we have 3 locations where you can do organic chemistry. We have Ångström Laboratory, BMC, and medicinal chemistry. And in Stockholm, we have 2 universities, Stockholm University and the Royal Institute of Technology, both with organic chemistry departments. And then here in the south, we have Linköping University, and to the west, we have Gothenburg, that carries 2 universities, University of Gothenburg and Chalmers. And all the way to the south, then we have Lund University. And that brings me to the presentation of today's speakers. So first off, presenting is Belén Martin Matute at the Department of Organic Chemistry at Stockholm University. She's going to talk about catalytic CH bond functionalizations and activations, mechanism and applications to late-stage functionalizations. And after Belén, we have Eric Corell at the Department of Chemistry, Umeå University. He's going to talk about chemical regulation of gene expression via small molecular interactions. And after Eric, we have Esther at the Department of Chemistry, Uppsala University. She's going to talk about reductions using divalent lanthanide catalysts. And after Esther, in the middle of the presentation, we have Joakim Andreasson at the Department of Chemistry and Chemical Engineering, Chalmers University of Technology. He's going to give a talk entitled Controlling Fluorescence in Photochromic Systems from On-Off Switching to Full Color Reproduction. And after that, we have Kenneth Wernemark from the Department of Chemistry, Lund University, who's going to talk about photoredox catalysis using charge transfer state of iron complexes. And after Kenneth, we have Daniel Strand from the same university, who's going to talk about asymmetric synthesis of ETP natural products from gleonitrine to collective synthesis of thiosilvateins. And last but not least, we have Philip Klan at the Department of Chemistry and Molecular Biology at the University of Gothenburg, whose talk is entitled Inspired from Nature's Design from Bioresponsive Tool Compounds and Tumor-Directed Migrostatics. And with that, I would like to thank you for your attention in this small presentation and give the word to Belén, who's going to come up next.
Thank you very much, Henrik, for this introduction. Can you hear me well?
Yes, yes.
Okay, excellent. Apologies. Okay, so I want to start by thanking you for giving me the opportunity to present the chemistry we are doing in my lab at Stockholm University. And today I will talk about primarily catalysis. We are a catalysis group directed towards organic synthesis. And over the years, we have used a variety of catalysts, both homogeneous catalysts, metal complexes, as well as some base catalysis. And also we have done a lot of heterogeneous catalysis using metal organic frameworks. But today I will focus primarily my lecture, my lecture on the homogeneous catalysis. I'm sorry, but I need to only check one thing because I have your pictures in the middle, so I don't see my presentation. Excellent, now I manage. Apologies for that. Okay. So a major focus in our chemistry is to develop catalytic reactions that can be applied to complex molecules. And therefore, when we develop new catalytic systems, we are very much focused on achieving very high selectivity, that the reactions may happen under mild conditions. We put a lot of emphasis on atom economy, and therefore we believe that they can be more applicable to real scenarios. And we have been focused a lot lately on applying the chemistry to the latest stage functionalization of complex molecules. We have also now started a program, well, it's been going on a little bit now, where we also want to develop the catalysis methods, thinking about sustainability as a priority. So we develop the methods by performing life cycle and toxicological assessment at the same time as we develop the methods. And I hope I can tell you about this in future lectures. So today I will focus on 3 areas using the homogeneous catalysis. The first one is on the activation of aromatic CH bonds and how we have developed this method for later stage functionalizations. Then I will also discuss how to use iridium catalysis as well for the reaction of slightly more activated CH bonds via alcohol oxidations. And then how we went from here to realize that sometimes we can just get rid of the metal catalyst and use a simple base catalyst for doing essentially the same type of chemistry. So for the latest stage functionalization, we wanted to focus on the directed aromatic CH activation of complex drugs. And the idea was that if we were able to selectively functionalize one of the CH bonds in a drug, we would be able to synthesize a possible drug candidate in a single synthetic step. So this is very important in drug discovery. So the problems or the challenges is that, as you all know, there are many CH bonds in complex organic molecules, but on the other hand, that means we also have plenty of opportunities to functionalize these molecules. The reason we were focused on the carboxylic acid as the directing group was because we have a parallel program where we were very interested in the functionalization of benzoic acids, as this can also be derived from lignin degradation processes. So we started first by looking at the model substrates, just simple benzoic acids. And the idea we had was that we wanted to use quite a simple catalytic system, and preferentially without the need of supporting ligands. Because if we were to extend this to later stage functionalization of complex molecules, we didn't want to end up in a situation where we would have to match the properties of the specific ligands with the properties and functionalities of a complex molecule. So this is how we came with this very simple iridium 3 catalyst. They have a CP start ligand and either the chloride ligands or the aqua complex derived from this one. So this is commercially available, the chloride, and you can make the aqua complex in a single step. This is very stable and you can store it for years without the special precautions. So with this catalyst in hand, we were able to perform the ortho iodination reaction of benzoic acids. And the beauty about this system was that it was essentially very simple. We only needed the electrophilic iodinating reagent and iodosuccinamide. We needed to use hexafluoroisopropanol as the solvent and the catalyst. So we did a screening of different benzoic acids, and the reaction worked very well in many cases. I don't want to go in much detail in the mechanism, but this is a typical concerted metallation deprotonation mechanism that involves iridium cycles. But I want to say something because it's important a little bit to understand what is the role of hexafluoroisopropanol, which we believe it has a double role. On the one hand is the solubility of the iridium complex. So it really works in this sense. But on the other hand, without hexafluoroisopropanol, the reaction just doesn't work. And by DFT, we determined that without hexafluoroisopropanol, the rate limiting step would be formation of the carbon iodine bond by a reductive elimination from an iridium-5 intermediate. But when we put hexafluoroisopropanol in the calculations, this is able to coordinate to this intermediate and remove electron density and therefore promote the reductive elimination. So with hexafluoroisopropanol, actually the rate limiting step is the CH activation. So it was key to use this solvent in this case. But a problem when applying iodination to complex drug molecules is that these are full of electron-rich aromatic, so we couldn't avoid electrophilic iodination reactions. So we moved forward, and this was the work of Eric Weiss, who was a PhD student in my group together with AstraZeneca. And so what he found is he could still keep using this very simple iridium catalyst with no additional ligands. But now we could use this boron derivative to install, instead of the iodide, to install this methyl group. And this is also quite important in medicinal chemistry because of the so-called magic methyl effect, which is a phenomenon that is observed in many drugs in a way that the pharmacological properties greatly are enhanced just by introducing a methyl group. So these were the first examples we did for the latest stage functionalization, these 3 drugs, where we obtain reasonable yields just in a single step. So AstraZeneca then studied the properties of some of these methylated derivatives. I have to say that you can also synthesize in the same way the deuterated analogs. And what they saw was that there was an increase in the metabolic stability for the drugs that they studied, and also that there was not significant differences between the deuterated and the non-deuterated analogs that we created or synthesized. So we moved forward, and then we decided to develop a method also based on the ortho-directed CH activation, but now to introduce the amino functionality. So we all know that amines are very important in medicinal chemistry, as the NH hydrogen bond is are the most common functional groups involved in fragment protein binding. So now we are using here an A site, still the same type of complex. In this case was the aqua complex also in hexafluoroisopropanol, and then we deprotect in situ. So we are able to synthesize the alpha-aminated benzoic acids, and the reaction can be a scale up at least to 9 millimole scale. And so Eric went on and also tested this in the latest stage functionalization of several drugs, and these are some of his results. I just want to highlight, for example, this one, fluorescein, where most likely this phenol here has to push electron density to break this bond, assist the ortho-CH amination, and then closes. And this molecule is obtaining 42% yield. So a good thing about having the amino group here is that also can serve as a handle for further functionalization. And this is what Eric designed. He used this functionalized A site so that it could be used as a feather to then further functionalize the molecule, and in this way created small molecule drug conjugates. So these are also some of the examples, and for example I want to highlight this one, which is formed in 63% yield from the commercial drug. So first is a CH activation to introduce this handled, and then deprotect the CBZ group, and then introduce this biotin derivative. And biotin is many cancer cells overexpress the biotin receptor. So this is one of the examples that Eric did. So with this, then we wonder, can we then extend this reactivity to other directing groups and not to be limited to the carboxylic acid derivative? So here what also Eric did was to identify what type of first screening conditions to be able to do this in very small scale by a high throughput experimentation. And then he screened many directing groups so he could identify which were good and which were bad. Then he also studied what was the outcome when different additives were added. So we could see what could be the functional group compatibility. And then with this information, he applied to several complex molecules and then very much the results were in line with his findings from the possibility of having different directing groups as well as the different functional groups that could be allowed and not allowed. So he created a predictive model. And these are two of the two examples that he predicted and then tested successfully. And in this case, we have this CH activation here next to the amide, and the drug was forming 72% yield. And this is another example where he used the pyridine as the directing group, also obtaining 81% yield of the functionalized compound. So this brings me now to the second part of my lecture, where we still keep very interested into developing catalytic methods for the synthesis of amines. In this case, we were focused on this reaction, which is the alkylation of amines using alcohols as the alkylating reagent. So this is very good because you do not need to use halides, and also you are able to stop at the monoalkylation. And also that the only byproduct produced in the best case is water. The price you have to pay is that you need a catalyst in order to activate the alcohol. So there are many catalysts in the literature that can do this chemistry, and of them, I would say iridium is perhaps that one that has been given the broader scope. And these are three examples by the group of Fujita, or this NHC iridium complex by Paris, and also the group of Kempe has done many contributions in this area. However, many of these catalysts still suffer from very similar limitations, which are high temperatures, they need to use base. Very often there is a large excess of the alcohol substrate needed. Some of them also have problems with dialkylation byproducts. And primarily the main problem, I would say, is the substrate scope, and is that the chemistry is limited to quite electron-poor amines, such as anilines. And this is because normally the amines inhibit the catalysis. So with this in hand, back already in 2015, we developed this catalyst, which is an NHC iridium-3 catalyst, which contains this alcohol functionality in the wingtip of the NHC. So this is the protonated under the reaction conditions, and the good thing is that this can then act as a proton acceptor, and the iridium acts as a hydride acceptor. So this bifunctional catalyst work really well in this model reaction. Very good yields. However, we didn't need a base. However, it was still limited to electron-poor amines. So we did some mechanistic investigations, and what we found was that after the alcohol oxidation, the iridium hydride is formed, and this iridium hydride gets inhibited, or it forms a resting state upon coordination of the aniline substrate. So this clearly explains why aliphatic amines, which are even more electron-rich, inhibit even further the catalysis. So we also saw that there was a dependence on the reciprocal initial rate of the aniline concentration. So we know the aniline coordination was an issue here. So what we did in this case was that to develop a second generation of catalyst that we could, with the aim of avoiding this type of resting state. And so instead of an oxygen, now we put an amino group. This is a better donor to the iridium, in a way that we thought perhaps we will not be able to break this oxygen-iridium bond, and therefore we would inhibit this the formation of such a stable resting state. So we synthesized this catalyst, and we tested it first with the model reaction, like anilines with different benzyl acid. So we use a small catalyst loading, and we could apply this chemistry for the alkylation of a large variety of anilines with electron-rich or electron-poor benzylic alcohols. And this was done for the first time at room temperature with no base and with an outstanding, or with a very good use of the reagents. So we only needed to use a 1-to-1 ratio of the alcohol to amine, and it was selective to the monoalkylation. So we, of course, tested more the substrate scope, and not only works with anilines, it also works with benzyl amines. with benzyl alcohols, with anilines, with different aliphatic alcohols, but also it works with different types of more aliphatic amines or even fully aliphatic amines. So we were very happy with these results, but we wanted to apply to more complex molecules and decided to focus on applying the method for the synthesis of non-natural amino acids. So what we wanted to do was to use a fully unprotected amino acid and see if we could introduce selectively one R group on the nitrogen. And the challenges here were that the alcohol, the amino acid was not protected. So it's a very rather polar zwitterionic substrate to work with. And also an issue here is that the reactions must be quantitative, because if the reaction is not quantitative, you end up with a mixture of the monoalkylated, perhaps even the bisalkylated, the starting material. In order to separate that, you have to go and have a protection step, purification, and then the protection. So this is definitely not good for the atom economy of the reaction. So we tested our catalyst and we found that it was really good for the alkylation of amino acids. And we tested more than 100 examples. This was work primarily done by Aitor with the help of Maiken. And more than 100 substrates. In all the cases, the reactions are quantitative. We can also keep, by doing some screening of the catalyst structure, we can also obtain the products in very high enantioselectivity. So these are other examples where we have changed the nature of the alkylating alcohol. And in all the cases, again, we see quantitative yields and also full selectivity for the monoalkylation. We have also been able to apply this to not only alpha amino acids, but also beta amino acids, and also to some small peptides with great success. And also what is interesting about this iridium complex is that it is fully recyclable. It is recyclable. So this is at least for 6 runs where we were changing the nature of the amino acid from one run to the other while using the same catalyst. And we in all cases still have the essentially quantitative reactions. The reaction can also be a scale up. And the solvent can also be recycled and reused if needed. So we have also focused on other type of redox chemistry from alcohols, but instead of intermolecular, this is an intramolecular example where we have an allylic alcohol and also using still the iridium CP star catalyst, we can transfer this hydrogen as a hydrate first to the iridium, and then this hydrogen is transferred to this other carbon here. So overall, this is a functional group interconversion where we can go from an allylic alcohol to a carbonyl compound. But when we use this metal catalysis for these reactions, we have this iridium enolate intermediates, and these may be reacted with different electrophiles. And this is what we have taken advantage of, the reactivity of enolates that are formed in situ. And we've been able to use many different electrophiles. This is one of the cases where we use chlorosuccinamide to synthesize these alpha chlorocarbonic compounds as single constitutional isomers. So the carbon chloride bond is exclusively formed at this carbon, and we don't see any functionalization at the other alpha carbon of the carbonyl compound. So these are very important intermediates also for synthetic organic chemistry. We have used not only chloride, but also fluorides and iodine and bromide and even oxygen. A problem with this chemistry is that we rely then on the use of these electrophilic species. So we wanted to instead using electrophiles to be able to couple these catalytic enolates with different nucleophiles. So we needed a polarity inversion of the intermediate, of the enol intermediate. And so we came up with this idea that if we add some iodine-3 reagent to the reaction mixture, we would be able perhaps to form these enolonium intermediates in situ, which are now electrophilic and can be reacted instead with nucleophiles. So the idea was now to be able to react allylic alcohols, which are readily available with different nucleophiles and the iridium catalysis to form these alpha functionalized carbonyl compounds as single constitutional isomers. So this worked very well when using methanol as the co-solvent. So we could synthesize these alpha mesoxycarbonyl compounds, and this was the iodine-3 reagent that worked best under our reaction conditions. So we could apply this to terminal allylic alcohols, but also internal allylic alcohols, slightly more functionalized allylic alcohols. And also we could use as the nucleophile this oxygen atom here when having these functionalized allylic alcohols. So this oxygen ends up having forming a carbon-oxygen bond on this carbon, giving rise to these furanones. The reaction also works when instead of a ketone here, we have an amide forming these functionalized furanones. So this brings me to the very last part of my presentation, where we realized in these isomerization reactions of allylic alcohols that when the alcohol was rather electron poor, we could just, instead of moving the hydrogen as a hydride, we could move it as a proton. And so we found that, Samuel in particular found that by using this guanidine type base, this TBD, we were able to deprotonate this CH bond here, and this is then added to the other end of the allylic system, forming also these carbonyl compounds. But what it was very interesting here was that when we had chiral allylic alcohols, we were also able to transfer the chirality in the type of allylic alcohols we were using were those having this trifluoromethyl group. So this gave a method for the synthesis of beta trifluoromethylated chiral carbonyl compounds. So these are some of the examples. The good thing about not needing a metal catalyst is that we don't need an alcohol here. We can have an ester here, such as in these two examples, and we could even have a chloride here. And it also works with a very high chirality, excellent chirality transfer in all cases. So the mechanism for this reaction is shown here. So the base deprotonates the CH bond, and this is the key intermediate. It forms this ion pair with induced non-covalent chirality. So it's very important that this ion pair it's kept tight so that the chirality transfer is very efficient. So if we, for example, use very polar solvents, this ion pair will not be tight, and then we would lose the chirality transfer. So we also, in the last more recent work, we have applied this also to allylic amines using the same TBD catalyst. And in this case, we have trapped the imine enamine intermediate with this acyl chloride. So we isolate these chiral trifluoromethylated enamides. And these are very important compounds also for further functionalization, as we have demonstrated in our recent work. So with this, I would like to finish with very simple conclusions, and is that sometimes quite simple catalysts are very efficient for the complex selective synthesis of very complex organic molecules. And I want, of course, to thank those that have done all this work, which is my research group. I'm very thankful to all the past and the present members of my group. These are the pictures of those that have primarily contributed to the projects that I have presented today. I'm extremely thankful to my collaborators, particularly Dr. Magnus Johansson from AstraZeneca for a continuous support over several years, and Perula Norby, Fami Himo, and Enrique Gomez Bengoa for support with DFT calculations. I'm very thankful, of course, for the support, financial support for our research. And here I show you also another beautiful picture of Stockholm, and inviting all of you to come and visit us. So this is all from my side. Thank you very much.
Great. Belen, thank you very much. We're off to a great start. Super talk on homogeneous catalysis. And true to form, first question comes in the chat from Perula Nurbi. Hi, Pio. Hope all is well. And the question is, or the comment, the critical HFIP, hexafluoroisopropyl, is, I believe, a PFAS, the perfluoroalkylate substance, and may be prohibited in a few years. Is anyone looking into equivalent alternatives that will not count as PFAS?
Yes, we are definitely looking into this very much. So we have tried to substitute because I still believe in many of these projects it has two roles. One is solubility. So in the second case, I didn't say in the NHC iridium complex, we have these cationic iridium complexes. So, and I believe there is a major role is the solubility of the complex. In other cases, as I said, it's just withdrawing electron density from some intermediates to promote specific steps. So we've been testing using other solvents, polar solvents, and then added tiny amounts of hexafluoroisopropanol. So in that case, we go at least in the right direction, but also some other benzoic acids. And we have seen some positive results, but they are not so far as good as these ones. But we definitely need to move forward. We can recycle the solvent so far, but I don't think that's the solution. We need to fully replace it. I agree with Perula.
Great. Thanks, Belen. And just to explain to panelists, you can just simply raise your hand and then it'll be very obvious who you are and you want to ask a question. But for those who are attending, can I ask if you either use the questions and answers box in your Zoom webinar, or you can raise your hand. And when you raise your hand, I can allow you to ask the question live. So that's the way it works on these webinars. So any further questions from any panelists? Let me see. From Frederick Dombrowski, this is a question in the Q&A. Regarding the recyclability, how do you win back the catalyst?
Yes, so in this case, it's a great advantage that the amino acid falls out, it precipitates. So then it's just a simplified filtration. And actually, there's amino acids alkylated. You don't need to do any column chromatography after. You just filter off and wash them. So you get rid of all the iridium and then you reuse the same solution where the catalyst is, and it is active. It doesn't lose any of the activity. And these reactions are run at the open air. It's a very robust catalyst.
Great, thanks, Belen. And there's a hand up from Johan Kayanis. Sorry now if I mispronounced that, but I've clicked the button allowing you to ask the question live. Johan?
No, sorry, that was a mistake.
That was a mistake. Okay, an early morning slip. You need to get another cup of coffee.
Yes, please.
Okay, thanks, Johan. Okay, any further questions? Seems not. So, Blen, perhaps in the interest of time, we'll stop there. Thank you very much, and I'll pass now back over to Henrik.
Thank you very much.
Cheers. Thanks, Belen.
Thank you very much, Pat. Okay, so the next speaker for today is going to be Eric Correll from the Department of Chemistry at the Umeå University. Eric, are you ready to share your slides?
Yes, I hope I'm sharing. Can you see the slides?
We can see the slides. Thanks, Eric. And we can hear you.
Okay,
and
you can hear me. Excellent, good. All right, so my name is Eric Correll and I work at the Department of Chemistry at Umeå University in the northern part of Sweden. And I would like also to start with thanking the organizers for inviting me to give this presentation. And then I would also like to say that I recently presented a similar presentation at the Medicinal Chemistry conference. So if you attended that, then congratulations, you now have the chance to repeat and get a deeper understanding of the topic. Or you can go for a second cup of coffee or something. So anyway, the title of the talk is Chemical Regulation of Gene Expression and how we can use small molecules and study their interactions with the genome to tune expression of certain genes. And understanding of our genome is of course interesting because this is related to who we are and how we act and react, right? So it's really interesting from a basic research perspective to understand how it is regulated and how it functions. But it's also, of course, have a therapeutic interest, because if we look at our genome from a therapeutic perspective, we can see that we only transcribe about 90% of the genome into RNA. And moving further, only about 1 to 2% of that is actually translated into proteins. And if we look at the drug targets of those proteins, it's only about 15%. And that is if we look then at the pharmaceutical drug being a small molecule and the target being a protein or an enzyme. So this means that we have a lot of potential of other targets, both as proteins, but also as RNA and DNA that we have not really explored that much. And now many of you maybe, of course, know that we have a lot of new work in this area in degrading proteins, for example, using PROTACs or molecular glues. And also in the RNA field, we have a lot of new development in terms of therapeutics, mainly these oligonucleotides therapeutics like the antisense oligonucleotides and siRNA. And this is really a field that has developed a lot over the last years. The first oligonucleotide that was approved for medical use was in 1998, and since then, the last years we have seen a lot of increase in this area, which I think is really interesting. We have close to 20 approved oligonucleotide drugs and many hundreds in clinical trials. So this is clearly an area that is developing a lot, and we see also that they can be used in many different therapeutic areas, which I think is interesting. And mainly the mode of action of these oligonucleotides is inhibition of the expression of the RNA then, so degrading RNA. So this is clearly an interesting area that can be further developed and likely will be. But if we take a step even further back from the RNA and look at the DNA, I think less developments have been done in the last years, and we know that we can have drugs targeting DNA. Of course, the intercalating drug, the alkylating and methylating agents, for example, they have been used a lot and are still saving lives today. But these are really invasive strategies. I mean, they kill cancer cells or kill bacteria, depending on their targets. So, of course, you will have a lot of side effects by using this kind of molecules that unspecifically bind to DNA. You know, in just one cell in a human, there is a lot of DNA. So if you stretch it out, it's actually up to 2 meters of DNA. So it's a lot of DNA in one cell, and that means, of course, that if you have an unspecific binder, you will have a lot of different targets for that molecule just in one cell. If you look at an entire human, then you have so much DNA, so you can actually reach the sun and back 500 times. So it's a lot of DNA in one human, meaning that you have a massive amount of targets if you target DNA unspecific. So I think for this approach to be viable, we need to find some specificity and to avoid side reactions and side effects of therapeutics. And so in my lab, we work with a specific DNA structure called G quadruplex or G4 DNA. It's a 4-stranded DNA structure that is formed in a single when you have a strand that contains a lot of guanines. So this can then fold up into a G4 structure. So one strand from the DNA can fold into this G4 structure. And in this structure, you have 4 guanines binding to each other in this format, where you have then 4 hydrogen bonds to each guanine. They are further stabilized by a central cation, and this form then called the G tetrad, then stack on top of each other to form this G4 or G quadruplex DNA structure. And if you compare this to double-stranded DNA, it's actually normally more stable than a double-stranded DNA. And the reason for this is that double-stranded DNA are composed of the DNA bases have 2 to 3 hydrogen bonds, whereas in this case we have actually 4 hydrogen bonds to each guanine. If you look at these structures in a surface perspective, you can see here you have the guanines in surface view in green, that they form almost stack on top of each other to form this pore-like structure, and you can see from the side that each of these G tetrads are really planar, and you have this cation sitting in between the planes. So what's the function of these structures? Well, they are highly enriched in certain regions in the genome. We have 700,000 potential structures that can fold in the human genome, and they are evolutionary conserved in specific regions, and they have important functions in replication and in the transcription of the DNA, and they are enriched in the promoter regions, so many genes where they can then tune the expression of these genes, and they're linked then to many different diseases, neurodegenerative, cancer, viral infections, inflammation, and so on. So we have a lot of different therapeutic potential with these molecules, with these G4 DNA structures. But this has, of course, resulted in a lot of interest in developing small molecules that can bind and stabilize these structures, because that would then prevent expression of the oncogene, for example, that is related to it. This is one example called FMDC3. It's a molecule that can bind and stabilize these DNA structures. But the problem is that we have so many G quadruplex motifs in the human genome, right? So if we need to find specificity, we need to also target these loop regions or the surrounding DNA, because just binding to this surface here will only will generate specificity between G4 DNA and double-stranded DNA, but not between different G4s, because they all share this same motif. So we have used different strategies in my lab to try to reach this specificity between G4 DNA structures. The first idea we used was structure-based design, and we worked with these indol structures and developed those that can bind efficiently to G4 DNA, but we saw that when they bind to the G4, they have this planar structure and this crescent shape structure. But in reality, if you do an energy minimization of this structure, they instead form more a planar structure, which is then not favorable for G4 binding. So we thought that if we lock it in the bioactive conformation, it would maybe increase the binding affinity of the molecule, but then the molecule becomes too rigid and it's hard for it to bind to the G4 structure. So what we next thought to do instead was to make this into a macrocycle, because if we make a macrocycle, then they are known to be efficient in binding to flat surfaces, and we will still also retain some of the flexibility of this molecule. So we did that by using the same bis indols. It was a lot of synthetic development to get to these compounds, quite complicated but my PhD student, Måns Andreasson, was talented and managed to succeed in this. We could see that when we macrocyclized these indoles, we locked them in the correct bioactive conformation, which improved the G4 binding and stabilization of the G4 structures. And we also get a very high selectivity for G4 DNA over double-stranded DNA because it's locked in this conformation. It's easily visualized in these more simplified molecules where if we lock this number 16 here into a macrocycle, you see an increase in the G4 binding stabilization. So we further investigate this in a bit more detail and changed. We saw that this indole is probably not needed. It might even be too big. So we reduced the size of the macrocycle. We also introduced a simple phenyl to compare, and we also replaced this methylated quinolines with just a benzofuran system. And the idea here was to see which parts of these macrocycles that were important. And so what we could see was that the move to the tryptamine was very good. We got quite potent molecules in binding G4 DNA in the nanomolar range. They had quite a planar conformation, and they were really electron deficient. And when we moved to this, replace the tryptamine with phenyl, we lost some of the binding affinity, but the molecules were still really electron deficient. And we think that the reason for this was that they now have a much more bent conformation and instead start to self-stack, bind to itself. And if we move to the benzofuran system, we lose all activity, and it has a lot of self-stacking, and it also has a very energy-rich system. So what we can conclude from this was that macrocycles are efficient in when you're designing new G4 ligand, G4 binding compounds. The reason for this is that they pre-organize the molecule in the correct geometry for binding to G4 DNA, and that an electron-deficient aromatic core is essential to engage in strong arene-arene interactions with the G4 surface. I don't show you all the details, but we have further studied this also in additional follow-up papers here. So the electronic properties combined with this pre-organization of the geometry is really important for a specific G4 binding. However, the problem here and the main goal that we wanted to achieve was to find specificity. And the idea was if we can bind strongly to the G4, and then we can try to find the later interactions with the side chains surrounding it to find specificity. But we were not able to achieve this and haven't been able to achieve specificity between G4 DNAs using this approach so far. We have also used a different approach where we instead use small molecule screening to find compounds that then are specific. And then we worked with this Swedish infrastructure, chemical biology consortium in Sweden, that can do screening of thousands of small molecules. And then we used 4 different G4 DNA structures and screened a library of small molecules to find compounds that specifically bind to certain G4 structures and not to others. And from this, we identified one of this structure here as one hit compound. We have worked quite some with this and made developments of these compounds into this second generation compound. And it's binding efficiently to G4 DNA with KD around 200 nanomolar. And we could also show that a little bit how it's binding, that it's binding on the top as we expected it to do. And we could also show a lot of biological details of what happens if we treat cells with this G4 binding small molecules. It has a very strong effect on cancer cells, for example. However, this was all very nice, and it resulted in nice stories related to G4 ligands and how they bind and what biological consequences they have. But still, we were, even though we screened for compounds with specificity for a certain G4 structure, we were not able to find this specificity. So we now used both screening for specificity and this structure-based design, but none of these have been successful so far. So it seems like it is really problematic to find selectivity between different G4s using traditional medicinal chemistry approaches. So what we're now looking into is a completely different approach, and that this is based, if we go back to the structure that we have, that G4 is formed, and then we have this flanking sequence. So then we thought we might be able to target or use this flanking sequence and conjugate our G4 ligands to a oligonucleotide that then could guide the G4 ligand to the correct position in the genome. And the idea then would be that we can guide it into a promoter region, for example, in an oncogene, and then bind specifically to that G4 DNA structure and then tune the regulate the expression of this gene. And so we did this, and the first thing we needed to do was to introduce or tune our G4 ligand a little bit and introduce this A side group on it. And using this A side, we then made a click reaction to introduce the guide oligonucleotide sequence. And when we tested these first derivatives, we could see by NMR that when we have the G4 DNA structure alone, these signals from the iminoprotons in the G4 DNA is very distinct. You have them here between 10 and 12 ppm, and double-stranded DNA comes between 12 and 14. So we can distinguish between G4 DNA and double-stranded DNA using NMR. So we saw that if we added the oligonucleotide alone, we formed double-stranded DNA, but we retained this G4 DNA structure, as you can see in this one figure here. And when we added our GLO, we call them GLOs, G4 ligand conjugated oligonucleotides, we could see that we still formed the double-stranded DNA, and that we had an effect on the G4. So meaning that the G4 was affected by something binding to it, and this effect was actually similar to the G4 alone binding, which is not part of this figure. And so we further studied this by binding using microscale thermophoresis, and we saw that the oligonucleotide had a strong binding affinity. The G4 ligand had around 200 nanomolar binding affinity, and if we combine the two, we actually had a synergistic effect where we had the binding of 12 nanomolar to the G4 with the flanking sequence. So what we could see was that this seems to be working. When we conjugate the G4 ligand to the oligonucleotide, it's still able to bind to the DNA to form double-stranded DNA, and it does not prevent the binding and the G4 ligand to bind to the G4 and stabilize this. So this was all good. What we next wanted to do was to test this in a more biologically close system. We used this assay called DNA polymerase assay. In this, you have a DNA polymerase that synthesizes DNA from a template strand, and if we then introduce a G4 structure in this template strand, the polymerase will stop at the G4 structure. But if we don't have anything stabilizing the G4, it will eventually be able to synthesize all the way up to the full-length product, and we can see the full-length DNA product up here. And if we have then a G4 ligand that bind and stabilize the G4 structure, you will see no formation of full-length product, and instead you will see an accumulation at this at the start of the G4 structure, which is then these nucleotides here. And you can have a control compound; it shouldn't do anything. And of course, we can have a template strand that does not form a G4 structure. Then the compound shouldn't affect the full-length amount of full-length product. So we used this assay and looked only on the full-length product here with our GLOs, and we could see that when we had the GLO with a matching sequence, we had a strong effect. Even at a very low concentration, it blocked formation of the full-length product because it stabilized the G4 structure. When we have a mismatching flanking sequence bound to the G4 ligand, then we don't see an effect. Almost no effect at all. The G4 ligand alone has an effect, which we knew from before, but it's not as strong as the GLO, which was nice to see. And it's not as you see also that when you conjugate the wrong sequence to G4 ligand, you actually lose activity from the G4 ligand binding. And if we switch, so we have a different guide sequence on the oligo on the G4 ligand, then of course the mismatch is turned around. So now here you have the red sequence being instead not able to bind. So this was very nice and shows that of the G4 ligand, the oligonucleotide is really guiding the G4 ligand to the correct position. I also show this because we are chemists, so we draw this how it actually looks like in ChemDraw to get an idea. And because you can think that, oh, but why is maybe the G4 ligand binding worse when it is conjugated to a oligonucleotide? But you can now see clearly that the G4 ligand is really tiny compared to the oligonucleotide conjugated to. So it's really a very small part actually of the entire molecule that is the G4 ligand. So we also looked at this by binding again, and we saw a very strong difference between when we have the correct and the mismatching sequence, which was very rewarding. And we further challenged this by using an assay where the same DNA polymerase stop assay, but we added both two different sequences, two different G4s at the same time, one with a correct flanking sequence and one with a mismatching flanking sequence. And then we had two different fluorescent readouts on the two. So we can look at the same gel by two different fluorophores, and then we could see which of the sequences that had been synthesized. And using this assay with the G4 ligand alone, we don't see any selectivity between the two different sequences. So it's unspecific, which we knew from before. But when we now add our GLOs, we see a clear selectivity for the target sequence. And so this was really rewarding, and this is something that we are now working on to further improve. We are looking now at the different G4 ligands and the importance of those. We are working on different oligonucleotide modifications. As I mentioned in the beginning, there's a lot of developments in oligonucleotides as therapeutics. So we could use this to get these oligonucleotides to enter into specific cells, and we can have them to get higher binding affinity, to get stability, and so on. So this is something that we're exploring now, and of course we're testing this in cells together with collaborators. I see that my time is almost up, but I'll just quickly say that we're working on having work on the linker here, where we have studied the different conjugation methods, the different lengths and different flexibility of these linkers. So strain-promoted click reactions and amide couplings and so on. And we studied how this affects the binding stabilization, and also try to model this system in moving away from the ChemDraw structure and actually doing some simulations of how it would look like when it's binding, to get a better idea of the entire system. So this is soon to be published. And so just to summarize, what we're aiming for here is to find specificity, small molecules that selectively and specifically can bind to one G4 DNA structure in the genome, and thereby being able to tune the expression of that particular gene. I mean, this would be super interesting if we could do this and then regulate, both study the function of individual G4 structures by selecting the correct guide sequence on the G connected to the G4 ligand. It would also open up the possibility to tune biological processes that are regulated by G4s. And of course, it would be interesting also to see if it has some therapeutic possibilities, because if you think about double-stranded DNA, you actually only, if you can find specificity to one G4 structure, you only have 2 targets per cell, right? So that's quite a big upside compared to traditional methods where you have a lot of many targets per cell. So you only need basically 2 molecules to enter the cell and to have an effect. I would also like to acknowledge people that has been involved in this, postdocs and PhD in my group. I already mentioned Måns Andreasson, who have done a lot of the synthesis. I have Alva and Sakina and Justina working on the conjugations, and Stephen also involved in this, and Rabindra. Collaborators, Shurd van Roy and his group, Mara, Andreas, and Namrat, have working closely together to develop this. Roger Strömberg has also been involved in initial work with the conjugation of these GLOs. And funding to support this, really important. Thank you for that. And also infrastructure support. And with that, thank you for your attention and open for question.
Great, Eric, super stuff. Very interesting. Any questions there from anyone in the audience, either as a panelist or an attendee? I don't see any. I suppose from my point of view, very interesting work. And you know, once you got the G4 ligand part of it kind of right, it was then all about using click chemistry to put on the, as you say, the very big oligonucleotide. Sorry, it's not my field, so I'm asking perhaps a very stupid question. Does it take long to find the right oligonucleotide, or is it obvious which one you should choose?
It's not a stupid question at all. I mean, I think that's something we're looking into. I mean, it's easy to find. The genome is known, right? So we can see where we can form a G4, and it's also been a lot of work on which G4s that are formed connected to certain genes, and so it's easy to find G4s that we can target, and then it's just to select the sequence that is banking that G4 and use that as the guide DNA sequence. So that is pretty straightforward to find one sequence that you can use, but how to tune this and what the best sequence to choose is to find specificity. That is, of course, something we need to look into and might differ between which G4 you're targeting.
Yeah. Oh, very good. Any further questions? Thanks, Eric. Any further questions from panelists or attendees? I don't see any questions coming through in the Q&A. Or from the participants. And how are we doing time-wise? I think we're right about smack on time. So Eric, perhaps we'll thank you for there at that stage. We'll thank you very much again for a super presentation. I love that JACS 2024 paper, really interesting stuff going on there. Again, not my field, but I can appreciate how good it is, so well done. And with that, perhaps I'll pass over to Henrik, who will introduce the next speaker. Esther. So over to you, Henrik.
Thank you so much, Eric, for a lovely presentation. Excellent work. Next presenter today is Esther Borbas from the university, from Uppsala University. She's gonna give us a talk on reduction reactions using divalent lanthanide catalysts. Esther, over to you.
Thank you very much. Does this, no, it doesn't look good. Or does it look good for you? Or do you see the next slide as well?
We see the next slide as well.
Okay, I'll just fix that. Sorry.
That's all right.
And while I mess around, I'd just like to thank everyone for giving me this opportunity to share our recent results on developing lanthanide-based reduction reaction. No, it's still the wrong one.
No, that's fine. That's good.
It's fine.
That's good.
Is it fine?
Yeah, from what we see, I just see the one big slide. That's it.
Perfect. Thank you very much. Okay, so yeah, I'm super excited about this opportunity, and thank you very much also for accommodating my time restrictions. I'm going to take part in a PhD defense in the afternoon, so unfortunately I will have to leave pretty soon. But thank you for letting me speak in the morning. So the background to this synthetic project really comes from just fundamental photophysical work. We've long been interested in understanding and using the luminescent properties of lanthanide compounds. Specifically, these are the trivalent lanthanides, so these are +3 charged ones. And many of the lanthanides have some sort of luminescence, whether it is in the visible, like europium, which emits red, or terbium, which emits green, dysprosium emits yellow, or in the near infrared, like neodymium, ytterbium, or erbium. These luminescent properties have found a lot of various applications. For example, if you look at a euro banknote under UV light, what you will see is that it's very colorful, and the red comes from europium. So I would like to propose that whoever suggested that we should use europium to make a safety for the euro banknote deserves a rise. The green one comes from terbium, and actually the blue one also comes from lanthanide. So there are applications in encryption, there are applications in light-emitting devices, and there are applications in biological detection. So these wonderful colors arise by emissions between 4F states, and the population of these 4F states is actually forbidden. So the way we normally do this is we stick a multidentate ligand around the lanthanide to keep it nice and stable, and we put typically an organic chromophore, which I will either call chromophore or antenna interchangeably, close to the metal. And what that does is it can absorb light very efficiently, so it generates its singlet excited state. Then it undergoes intersystem crossing to give its triplet state, and the triplet state will participate in energy transfer, hopefully very efficiently, to give you the excited state lanthanide. And when this excited state lanthanide goes back to the ground state, that's when you get these beautiful photons, the color of which is typical of the lanthanide, which is of course great if you're trying to detect it in a complex environment. So this looks wonderful, and we've been really attracted to these. And one of our motivations was to develop responsive probes. This is a very old story, so I'm just going to summarize because really the problems that we have encountered here set the stage for everything else. So we made responsive probes that, in the absence of their target analyte, which could be a metal ion, it could be an enzyme, it could be a reactive small molecule, were non-emissive. So you had this lovely dark background, and depending on what this caging group was, the analyte would cleave the cage chemoselectively. You would get a cyclization to form the antenna, and you would get a turn-on response. And the only reason I'm mentioning this, I wasn't even going to include this slide, but I noticed that Alison Holm is sitting in the audience, and I just wanted to tell her that we've been really inspired by her work on copper responsive probes. So that's great to be able to say now. Now, we were very happy with the versatility of these complexes. One thing we were not happy about is that even the turned-on probes were, even though they were giving us a good signal over the background, they were not very emissive. The quantum yields were really poor. So what we started to do was to look into what was depleting our excitation energy. So I showed you a very simple sensitization graph. The problem is that if you start to look into how sensitization actually takes place, what you discover is that there are a lot of things that can go wrong. So at every single stage, there are processes that can quench your compound. So for example, the singlet excited state of the chromophore, the antenna, can of course emit light itself, fluoresce. It can undergo collisional quenching, or it in its excited state, it can be a better oxidant or reductant than in its ground state, so it can participate in electron transfer. Of course, the triplet state is again amenable to quenching by, for example, atmospheric oxygen, and the lanthanide excited state itself can also participate in a number of processes, of which XH quenching, so quenching by the solvent oscillators, has been really well studied. So what we wanted to know was what was really doing which one of these processes was really depleting the excitation energy of our complexes. So we did a structure activity study. We made a whole bunch of complexes with similar structures. So we kept the binding site constant, and we did a huge variation in this antenna structure. And I'm just going to show you one because it's very typical of what we were seeing. So what we saw was that the lanthanide-based quantum yields, they varied a bit. Some of them were pretty good. Some of them were pretty bad. But the interesting thing was that, well, if we already recorded the entire fluorescence spectrum, we also calculated whatever the ligand was emitting, so the fluorescence that was coming out of the ligand. And when we put these numbers together, we found an interesting trend. The trend was that the least amount of fluorescence was coming out of europium complexes, then samarium, then dysprosium, then terbium, and then gadolinium. Now, the fluorescence of a ligand of the chromophore can be depleted by a number of processes, but you wouldn't expect this to be constant, or the trend to be constant across a whole range of organic chromophores. So you can, for example, have singlet-mediated energy transfer to terbium, dysprosium, or samarium, or europium, but you wouldn't expect that to be exactly the same for every single one of these chromophores. But what we noted was that, well, this order, europium, samarium, dysprosium, terbium, gadolinium, actually terbium and gadolinium are at the back, is really the order in which the +2 oxidation states of these ions are stable. So europium-2 is relatively accessible. In some complexes, you can even make in water. Samarium-2 is, of course, a very well-known organic reductant, single electron reductant. Dysprosium-2 is a little bit more difficult to make, and terbium-2 and gadolinium-2 are really not very accessible at all. Terbium-2 can accept singlet-mediated energy from the antenna. Gadolinium has neither redox nor photophysical activity in these complexes. So we use that as a sort of dummy atom. It's a nice heavy atom with the same Lewis acidity, or very similar Lewis acidity as all the other lanthanides, but it's not going to do any kind of funky photophysics or redox chemistry. So we thought that, well, that's very interesting. It looks like, oh, and by the way, a 2.8% europium emission is not great. So it's like, okay, maybe what is happening is that we're having electron transfer, and the electron transfer is taking place to the lanthanide. And this is what is depleting our complex's excitation energy. What I'm showing here in this table are the reduction potentials of the +3 ions for the different lanthanides. And what you can see here is that in this block, I mean, these are really typical for transition metals, for which we don't normally expect. We expect redox activity to happen. So this is really not surprising. So how can we address this? Well, one way to address it is to make sure that the antenna is more difficult to oxidize, right? So we've done that. We've put fluorine groups, and we did quite a bit of synthesis on this. We walked it around the antenna. We also did similar things with the CF3 group. What we found was that, yes, in certain positions, electron withdrawing groups on an antenna can stabilize the +3, can slow down electron transfer from the antenna, and we can get reasonably good emissive properties. So, for example, this europium complex has a quantum yield of 5.4%, which is about twice as what we were seeing in the previous one, although it's not a huge improvement compared to the non-fluorinated one. The problem was that other things were also changing. For example, the triplet level of the antenna, which impacts the energy transfer from the antenna to the lanthanide. So even though some improvement was seen for the europium complexes, for the terbium complexes, in some cases, it was really detrimental. So I thought that, okay, maybe the antenna, we should just keep it as it is, but can we stabilize the +3 oxidation state of the complex? So can we just make sure that the europium doesn't want to get reduced? And our very naive idea was that if we're having carboxylates here, then formally we're having a zero overall charged complex. Now, if we're one by one replacing the carboxylates to carboxamides, then we go to a +1 charge, +2 charge, +3 charged overall charged complex. And we were expecting this +3 charged complex to be more amenable to reduction at the europium than the one that's zero charged. And we could, of course, check the stability of the +3 oxidation state of the europium in these complexes by looking at its redox potential. And what we saw was that irrespective of the antenna, yes, the zero charged complexes were having europium-3 of about the same stability. And as we were piling on the charges, the europium-2 was becoming more and more and more accessible. And when we studied the photophysical properties of these complexes, what we found was that the zero overall charged complex carrying the exact same antenna and having the exact same coordination geometry was up to 5 times more emissive than the +3 charged one. So we were very happy about it, because once you spent a lot of time fine-tuning the excited states of antennae, you really don't want to go around and then maybe mess it up by putting on electron withdrawing groups. So this is a good way of improving the emissive properties of a lot of europium complexes. And we also think that this is a nice way of maybe designing responsive probes. So lanthanide-based responsive probes have been based on modulating the coordination geometry of the complex by increasing the hydration state or decreasing the hydration state, by modulating the antenna, so making electron energy transfer more or less facile. We believe that there are, well, we haven't been really able to identify a complex where what is done is that responsive probes are being generated by simply modulating the redox potential of the metal. So this was a very interesting little experiment. Our complexes are octadentate, and europium in aqueous systems has a 9th ligand, which is a water ligand. Now, this water ligand is quenching, and everyone who works with luminescent lanthanides knows that that's not a good thing to do. Now, if you add fluoride, sodium fluoride or potassium fluoride to this solution, what you see is that there's a huge increase, and a 7.5-fold increase in the lanthanide lanthanide-based quantum yield, so you get a turn-on response. And it's very easy to say that, okay, that is due to the fact that you have replaced the water molecule. And we went actually a little bit deeper in the analysis of this system. And if you think about what lanthanide luminescence is a composite of, well, you have to populate the lanthanide excited state. So all the processes that go into exciting the antenna and the energy transfer can be described in this sensitization efficiency. So that gives you the efficiency with which you're creating an excited state lanthanide. And then the intrinsic quantum yield of the lanthanide, which is once I have the excited state of the lanthanide, how likely I am to get a photon out of its returning to the ground state. And you would expect the water molecule to show up here in the lanthanide coordination environment, while a redox process affecting the antenna and the lanthanide would be in the sensitization efficiency. And there are ways of getting these numbers specifically for europium. So what we found was that the 2.3-fold increase was coming from the fact that we replaced the water ligand, and the larger 3.4 time increase came from the fact that we shut down a photoinduced electron transfer in the system. So this would be very interesting to see if we could expand that further. But I would like to go in the other direction now. So instead of trying to shut down photoinduced electron transfer and getting better emitters, we were wondering if what we could do was to instead capitalize on this electron transfer. Because I have a background in organic chemistry, and the moment we started to see draw these mechanisms with lanthanide 2 in it, I thought that, well, maybe we can just intercept it, and this could be a way to generate lanthanide 2 reductants catalytically. And so we thought, okay, maybe we could do this, have a sacrificial donor here, regenerate the antenna, and then the substrate could intercept the lanthanide and give us a product. So I like to show this slide for motivation. Samarium 2 iodide, I don't know if everyone has worked with it. It's a magnificent reagent. It is just so wonderful. This is the largest scale industrial application. I believe it's still the largest scale industrial application, so it's not a great scale. But just observe this magnificent molecule with a whole bunch of very sensitive functionalities. And what samarium iodide does is it cuts out a single functional group and replaces it with a hydrogen. The problem is 1.27 kilos of the substrate need 21.4 kilos of samarium iodide, which is clearly not a sustainable thing to do. So it's a wonderful, highly chemoselective reagent that really should be used a lot more, except for the fact that the average stoichiometric reaction uses 2 to 10-fold excess of samarium iodide. So it would be great if we could do a catalytic version. So what we did was we designed a number of catalysts. And the way we designed them was that, well, we had the same sort of coumarin or carbostyrol type chromophores that we were using in our luminescent compounds, because we suspected that those were already undergoing photoinduced electron transfer. And then we put a ligand binding site that we got from the literature, because these were reported by Matt Allen to stabilize the +2 state of europium. This is the experimental setup. So we do small scale reactions, and then we can, of course, scale them up. And then we have L3, which is a carbostyrol. So we expect that electron transfer would take place. And maybe we were just expecting to get lucky, and maybe these coordinate sufficiently to give us electron transfer. Now, I'm not going to walk you through the entire, well, 1+ year of working out the details, but I'm very happy with these results. So this is part of the substrate scope. We started with the benzyl halide reductions, and in the presence of a sacrificial reductant, which could be zinc, just a strictly one equivalent of zinc, but it also can be diisopropylethylamine. Again, just one equivalent. In the presence of water, with 365 nanometer or visible light irradiation, what we could get was very selective reactions, either for the corresponding toluene or the dimerization reaction. And this is very interesting. So first of all, we have a very nice functional group tolerance. We can have aryl halides under these conditions. We can have sulfides. We can have ketones. We can reduce heterocyclic compounds. Depending on the conditions, we can either get the biphenyl or we can get the toluene. So for example, here, we can just choose the conditions to get either of these with high selectivity and good yield. If we replace the europium with samarium, what we can get is a more potent reductant, which means that the selectivity changes. On the other hand, if we're getting sluggish reactions, we can improve that by replacing europium with a samarium. So we can tune the reactivity by changing the lanthanide itself. In practice, this means you're mixing the salt. You just take a different lanthanide salt for the reaction. So we could also use slightly more forcing conditions and take these heterocyclic and aromatic compounds and reduce the aryl halides. We tried to make comparisons to either reported stoichiometric conditions or conditions that we have tried ourselves. So in stoichiometric conditions using 8 equivalents of samarium iodide and isopropanol, we got absolutely no product for the reduction of this compound, and we got a 50% conversion with GCM, as the isolated yield is a little bit lower because it's a volatile compound. And we could do other functional group reductions as well. So we could do a catalytic phosphate reduction. We could do NN double bond reduction. Reduce the azide to the NN double bond. Of course, the key here is that the electronic properties of these aryl groups are different. So this one goes to the full saturation while this one stops here. And this is one of my favorites. So we can take the paranitrobenzaldehyde, and under one condition, we get perfect selectivity for the reduction of the nitro group. With samarium iodide, there's none of this being formed. If we take the same catalyst and add water, a little bit of water, then what we get is very nice selectivity for the alcohol without touching the nitro group. And we can, of course, do other reactions as well. One of the great advantages of samarium reagents is that the context they are being used is you initiate a single electron reduction, and that then gives rise to further carbon-carbon bond formations. And we could do this as well. So we could do aryl and heteroaryl alkylations, and for example, in the synthesis of papaverine. So that's nice, but I've been drawing this little cycle, and you might have a very valid question of everyone can draw cycles. Do we actually have any kind of proof for the formation of either this species or this species? So we tried to do some control experiments and try to make sure that what we're doing is really an lanthanide-based redox chemistry. So this is our control ligand. So this recreates the coordination environment of the metal, but it doesn't have a chromophore. So we can excite it all we want with visible light, but nothing's gonna happen to it. So I mentioned very far back at the talk that gadolinium is a dummy atom with no redox activity or no photochemical activity. So when you see a gadolinium reaction, what that means is that we're doing a reaction with a non-redox active lanthanide. And if we replaced europium or samarium with gadolinium, under none of the conditions did we get meaningful amounts of the product, which means that the reaction does not proceed through the chromophore. So this is still the full ligand. So just the chromophore is not sufficient. Again, if we even take out the lanthanide, no reaction is taking place. If we have the fully assembled complex, but we do the reaction in the dark, no reaction is taking place. So we do need the photoexcitation of the complex. And if we take one equivalent of the complex with irradiation, but no reductant, then we get a very decent yield of the product. So what this experiment tells us is what we need the sacrificial reductant for is the catalyst turnover. It's not reducing europium in situ. It's not doing anything else. It's turning over our catalyst. And the control experiments with this ligand without the chromophore showed us that we do need the chromophore as well to generate the active species. So that was very, very promising. But we really wanted to get some proof for those. Well, first of all, for the antenna radical, the chromophore radical, and also the europium-2 species. So the first experiment we did was we irradiated the europium complex with the visible light in the presence of tempo, and we got the tempo adduct, which we could characterize. We also irradiated it in the presence of this radical trap, and we could take the EPR spectrum of that. I mean, this was sort of the icing on the cake, because we've been really desperate to record a spectroscopic signature for the europium-2. And what we found was that we could take this ligand and make a complex with chemically generated europium-2. And we could take the EPR spectrum of that, and we get the characteristic europium spectrum. And we could take the europium-3 complex of our catalyst, irradiate it, and again, get the EPR spectrum of a europium-2 species. There are some So we were very happy about it. This is direct evidence for europium-2 being formed under the irradiation. So what we're doing now is we're moving away from our current catalysts because we would like to have species that absorb more towards the visible. So recently, Monica Tomar, a PhD student who's going to graduate in December, has completed a study where she just took off the shelf a large number of organic chromophores. I mean, these are like fluorescein and rhodamine and coumarin 343, and some of these have very nice visible absorptions. And she investigated the abilities of these chromophores to generate europium-2. So again, both combining EPR spectroscopy and also UV vision luminescence spectroscopies, she could identify the spectroscopic signatures of europium-2. So this is one of the directions that we're going in. So with that, I would like to summarize what I talked about. So I began by introducing photo-induced electron transfer in luminescent lanthanide complexes. And I hope I could show you that this can be a significant quenching process. In fact, for some of the complexes, this is comparable to the very well-studied XH quenching. So this is something that might be worth addressing in europium complexes. And it's possible to address it either by making the antenna more resistant to oxidation, or by making the europium-3 compound more resistant to reduction. And I've shown such strategies for both. And then I also showed you that the transiently formed lanthanide 2 species can be oxidized by a whole range of substrates. And this is a good method to recapitulate the reactivities of stoichiometric samarium-2 iodide reagents with the sort of selectivity and scope is comparable to, and in many cases, even better than the stoichiometric reactions. The reactivity and selectivity can be tuned both through by simply changing the type of lanthanide you're using, but you can also change the ligand, or you can change the reaction conditions. And these reactions work at room temperature with visible light. They tolerate water. In fact, we have examples with just pure water working, and we can use mild terminal reactants and catalyst loadings as low as 2%. So with that, I would really like to thank the people who have done the actual work and the funders who had made this study possible, and I'd be very happy to answer any questions.
Super, Esther. Thank you very much. Very nice presentation on the use of lanthanides in synthetic organic chemistry. Great stuff. Now it's open for questions, and there's already one in the Q&A. And again, I'd remind both panelists, but more particularly attendees, if you wish to ask a question, you can pop the question into the Q&A box, or you can raise your hand and I can unmute you, and then you can ask the question yourself. So there's a question here from Dr. Adil. Are there any safety protocols when working with lanthanides?
So nothing in particular. I mean, you don't want to swallow them because they are toxic, but you don't normally do that with most reagents either. They're reasonably nice things to work with. They're a little bit hygroscopic, but that makes more the experimentalist life difficult rather than sort of dangerous. Of course, these are photochemical reactions, so you want to be careful with the excitation sources. But at least on this scale, there are no major concerns. I can imagine that if you're doing the stoichiometric reactions, yeah, I mean, releasing a kilo of samarium iodide, yeah, don't do that.
Yeah, no, good advice. Henrik, you also have your hand up there to ask a question, please. Over to you, Henrik.
Thank you, Esther, for a lovely presentation. Awesome work on the lanthanides. My question is, you showed early in your presentation that water is detrimental for the quantum yield, but later on you also use water sometimes to improve your reaction. How does that come together?
So water is detrimental to the quantum yield, but most of us still live with a coordinated water and a luminescent compound. But for the reactions, we don't actually worry about what's happening at the excited lanthanide because we don't want to make the excited lanthanide. So if you go here, the water is quenching the lanthanide at this stage, right? But when we're doing the reactivity, we're actually going in this path. And sure, you can imagine that maybe the singlet state of the antenna is also quenched by water, but it's not as sensitive. So the reactivity doesn't get affected, or in some cases it gets improved. And I mean, these are competing processes, and when we're doing the synthetic chemistry, we just don't want to go down this path. We just want to go to the electron transfer path.
Okay, thank you for a nice explanation.
Great. Thanks, Henrik. Any other further questions? Seems not. So perhaps in the interest of time, and we're smack on time, so that's really good. I would suggest maybe that Sweden's doing much better than previous countries on this forum, so well done. So again, we'll pause there if that's okay, Esther, and thank you again. And pass over to Henrik, who's going to introduce EuChemS, who's our next speaker.
Okay, thank you very much, Pat. Joachim Andreasson from Department of Chemistry and Chemical Engineering, Chalmers University of Technology, will give you the next presentation. He's going to talk about controlling fluorescence in photochromic systems, from on-off switching to full color reproduction. Sounds very exciting, Joachim. Looking forward to your presentation. Please go ahead and share your slides.
Joachim, hello.
Sorry about that. My bad. Can you hear me now?
We can hear you now. Yes. Your slide looks very small, at least to me.
All right. What if I...
Now it's disappeared, and I have some...
Yes.
Yeah. I mean, it's about only 20% the size of a normal slide, 20%, so it looks really small.
Yeah, I'll try to do the following.
Don't worry, this happens with live webinars all the time, Joachim, all the time.
Like so, and then is it the same problem still?
Yeah, I can't see anything yet.
Share and
Yep, that looks better. And if you just go to the now we can see the next slide as well. So you might want to change that. I think it was an Esther solved that problem. I'm not sure how that's done, by the way.
So let's see. And then share and screen one. Still very small, right?
I'm afraid so.
Ha, 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 ha. And share. Well, I don't really know what to do about this.
Esther, will you be able to help? Because I think you had exactly the same case when you started to present. Yeah, that's looking good. And press slideshow.
Sorry, just stop that for a second.
Yeah, no, you're okay, Esther. I don't know if you can help Joachim, but when he starts to present, the next slide shows up as well. So I think you had something similar, didn't you?
Yeah, I started the presentation and just shared the started presentation. So instead of opening the presentation in Zoom, I already started the slideshow separately.
Yes. Yes, that's what I'm trying to do here as well.
Good advice. Let's see if it works.
Well, maybe I have to unplug. I think it's
Yeah. I wouldn't suggest unplugging anything, Joachim. I don't know if that's going to help.
I think I will unplug one of the screens and then do it again. Like so, share.
Again, very small.
Again, very small.
I'm afraid so. Yeah, we still have the next slide coming up as well, so we can see the slide you're talking on and the next one.
Yeah.
So have you done what Esther suggested, that you start the presentation already in slideshow before you start sharing?
Indeed.
Yeah. Kenneth, over to you.
Can you see my screen now?
Yes. Yeah, perfect.
Okay. Thank you, Pat, and thank you, Henrik, for this invitation to present my research. And this is research that started around 12 years ago. And it has taken us a lot of time to develop iron as an efficient photocatalyst in photoredox chemistry. And we are about now to, so to say, make really good reactions working with iron. And apart from developing reactions, and we also want to make very thorough mechanistic studies in collaboration with other people at Uppsala University, for instance, and here at Lund University. Because as you will see, some of the mechanisms are a little bit different compared to using traditional noble metals for photoredox chemistry. So my talk today will be a short introduction to photoredox catalysis, an introduction also to iron photoredox chemistry, and also to photophysics that is very important for iron complexes. And I will have two showcases of photoredox catalysis involving iron chemistry, aminomethylations of electron-deficient alkenes, and the other example will be atom transfer radical addition reactions. Classical reactions, at least the last one in photoredox chemistry. The first one, we have a little bit of a twist, actually, that I think we are kind of alone with. And I will make some conclusions. So if we start from the beginning, where photoredox catalysis basically took off, and that this is to show actually the importance of changing, exactly like Esther was doing with her replacing samarium iodide. Here we are going to replace tin radiants in a very important organic transformation. And the traditional way to do that is taught in all organic chemistry courses, is that you use tin radiant and you use a compound that can generate radicals, either, as we will see, by light or by heating. And important here is that you need light of high energy, UV light, and that generates now a tin radical. And this tin radical attacks the anti-bonding carbon-bromine bond. And we generate carbon radical, and this carbon radical can abstract the hydrogen radical from the tin compound, and we have put down now a hydrogen instead of a bromine. The problem here is that tin is, of course, toxic and environmentally friendly. And people at that time, one of the pioneers, Corey Stevenson, considered if there any other way we can make the carbon radical than by this method, classical method. So what he did is that he used a ruthenium compound to do this, and this ruthenium compound is called a photocatalyst or photosensitizer. And what we're going to use here is what will be very important here is that we can make ruthenium-1 species. And to reduce to this reduction from ruthenium-2 to ruthenium-1, that will be facilitated by the photoredox potentials of the excited state. So this is the key thing in photoredox chemistry, that you have better redox potential in the excited state than you have in the ground state. So how do we do this? And of course, at that time, there was no LED lamp, but today I'm showing this with a LED lamp, and you see already now that we go from ultraviolet light to blue light. So we are actually improving this and going to more red light part of the spectra, so to say. And we make the excitation to the ruthenium excited state, and this ruthenium excited state can then oxidize triethylamine, that is the additional and sacrificial electron donor, and we make the radical cation of the triethylamine, and we have reduced now formally ruthenium-2 to ruthenium-1 via the excited state of ruthenium. And this ruthenium reduces the carbon halogen bond by putting an electron, as I said, in the anti-bonding orbital. We get the radical anion, and this radical anion loses an halogenoid, and we get the radical by itself. And of course, now we have converted this. Oh, sorry, I don't see this point. So sorry, I have to move something to a green process instead. And I will just, before moving on, I will talk a bit about the important part here in the photoredox catalysis that makes the difference, and that is the photosensitizers. And in this case, what they used is actually ruthenium tribipyridine complex, and by irradiation with light, you actually take electrons from ruthenium here and put them on the ligand. And that creates a charge separated state. We have a minus charge we get the minus charge on the ligand, and we get the plus charge on ruthenium. And this hole now that we have created on ruthenium is then able to accept electrons from a reagent, and that in this case is a donor. And we oxidize this donor, and the negative charge we have on the ligand can act as a reductant and reducing substrate, and that will be called here in the former case an acceptor. So we have done a reduction. And the advantage with the charge transfer state to use that as the photofunctional states in this reaction is that they put the excited states in contact with the surroundings, because we want to make a bimolecular reaction, and of course we will put the reactivity so it can be on the ligand that can actually get in touch with the substrate that is, of course, in the surrounding of the photosensitizer. And the charge transfer to ground state transitions are not spin allowed. So that means that we have a good chance that the excited state exists in such a long time that it can meet another molecule and do the electron transfer. So we get a long excited state lifetime by this. And charge transfer states absorb light better than metal center transition that often are photophysically forbidden. And they also appear at more panchromatic wavelengths, so we can absorb more light from the entire solar spectra than if you have, for instance, metal center transition. And also ligand design make it possible actually to design or set the lifetime and redox potential of the charge transfer state by making improvements in the ligands. And the charge transfer state as such then is a good state for to use in photocatalysis. But as we see, we have ruthenium, and the good thing with ruthenium, as you see here, it is a lifetime that is around a nanosecond, which is a very high excited state lifetime. But the drawbacks is that the metal is scarce. We don't have a lot of ruthenium, and if you go to another metal that is used a lot, we saw example of that is iridium. There is even less of that. We will see that, and also that it's expensive, like all noble metals. So, if we want to replace ruthenium, that we see here is one of the rarest metal on Earth, one could of course look then at iron, and we know from our textbooks in chemistry that we once say that compounds that are in the same group in the periodic table have basically the same physical properties. And iron then, we see here, is one of the most abundant. It's actually the second most abundant metal in Earth crust, constituting 6% of Earth crust. And we can, for example, of course, it could be worse than use ruthenium. We could use iridium, that is one of the very rarest metals on Earth. But the question is now, how can this be done? And I said that chemical and physical properties are very close for compounds in the same group in the periodic table. But when it comes to physical properties, it's a big difference. So here we have a typical ruthenium tris bipyridyl complex, and here we have the MSCT state that we're going to use as the photofunctional state, and the activation pathway that is possible is go to something called the triplet metal center state. And we see here that for ruthenium, the activation barrier to go there is rather high, and also the thermodynamic driving force is not favorable. So this deactivation pathway we don't have in ruthenium. Maybe if we heat it, we have a thermal deactivation to the MC state. And that's why the MSCT state, that is the photofunctional state, is of high site height lifetime in ruthenium. If we now look at the iron tris-bipyridyl complex, we see that this MC state is lying in energy, and that means that the activation energy here is much smaller, and also we have a thermodynamic driving force to go into this sink here. Unfortunately, the time is not enough, so I can explain on a more molecular level using ligand field theory and so on why this is the case. I want to instead move to the photocatalytic applications. But what about this? If we could now push up the metal center state, and we can get into something, situation that looks like this, that the MC state, of course, we have no ambition actually to put it over the MSCT state in ruthenium. That would maybe be too difficult. But at least to push it up a bit, and we see now that the activation barrier that was lower here in the traditional iron complex is higher. So we have the chance actually to have the MSCT state with a longer lifetime. But the question is how, and what should the ligand look like to do this? And we came to the conclusion, since my background is in thermocatalysis, that N-heterocyclic carbenes, they have this very good effect that they actually increase the metal, the electron density on the metal. And that allows, for instance, oxidative addition reactions of unreactive substrate to be facilitated. And this is exactly what we want. We want to have something that put a lot of electrons on the metal, pushing up the metal-centered state that is centered around the metal. And if you look at N-heterocyclic carbenes, we see that they do this very well by having good sigma donation. So here we have the carbene, and here we have the metal, and we see that we have a very good interaction going in the right direction. And instead, the pi back donation from the metal to the ligand itself is hampered, although we have an empty sp2 orbital here on this carbon. Because of the nitrogens that sits here, we have kind of electron donation from those nitrogens, making the orbitals here, the empty orbitals, at least partly filled. So in total, we have a net electron donation that is mainly sigma. And how do we realize this now? So here we have the iron 2 complex, and the lifetime, the traditional one, we have 100 femtosecond, and that has the MST, excited state lifetime. And unfortunately, it seems like I made the ground state lifetime of both of them. But the meaning is that one of those electrons should go up here in the pi star of the ligand. Sorry for that. And how do we realize this transformation? So we started in putting 4 NHC moieties in an octahedral environment, and we ended up with this complex that you see here up to the right. And that complex then we have a rise in the excited state lifetime of that complex, and we see that we have moved up actually the MC states here, and we have used in our collaboration femtosecond spectroscopy to map the excited state dynamics of these compounds. So we got actually a 9 picosecond lifetime, and this was in 2013. So we had actually increased the excited state lifetime by 60 times by exchanging bipyridine ligands for ligands containing N-heterocyclic carbenes. But this excited state lifetime is not enough to do bimolecular reactions that we want. Instead, we replace everything here with 6 NHC moieties, and then we got the scorpionate ligand, and it shows then that we have replaced, we have put so much electrons into the iron, so we actually oxidized iron 2 to iron 3, and instead of having an MST excited state lifetime, we have an LMCT, ligand to metal charge transfer, and here I made it correct. It looks like this doublet. And that is now the excited state lifetime. But the principle actually turned out to be the same, that we raised those metal-centered states here, and we got an activation barrier here, so we could get the lifetime of 2 nanoseconds. And that is enough actually to, as you will see, to do bimolecular reactions. And another thing that came out from this is that if we irradiated with green light this complex, we actually got photoluminescence back, red light photoluminescence, and this was actually the second example of having an iron complex being photoluminescent, and with a rather good quantum yield, 2%. And we could also reduce the excited state. Reduce the compound to iron 2, and that has an excited state that then will be MLCT, 9 picosecond. So we have oxidation state of both iron actually, and iron 3 and iron 2. Iron 3 has a rather long excited state lifetime. We will see if the iron 2 is needed at all. And we have absorption, as you see here in the visible region. Both oxidation and reductions are metals-based. That means that they are reversible, which is very good for catalysis and very beneficial redox and photoredox potential you will see here. Sorry. And if we now look at the spectra, we see that the iron 3 complex here, the iron 2 complex here absorbs at 350 nanometers. The iron 2 absorbs light at 500, and our photoredox reactions we will do with 530. So we will selectively be able to activate the iron 3 complex. And the first reaction I want to show you using photoredox chemistry is when we take a methyl amino compound where we can put the radical here, and we make a Giese type of reaction to an electron withdrawing substrate with a double bond, and we can actually then introduce a methyl amino group into that using this photocatalyst. And the key thing here is that our excited state here matches actually the oxidation potential of those alpha amino compounds, so methyl compounds, so we can make radical cations like this using the excited state. The problem is here that this radical cation that is generated could actually decompose to different compounds, and it's only this one we want. So to circumvent this problem, we actually used amino compounds using a methyl TMS group. And that radical cation that then is used, it will be stabilized by this beta effect of silicon, and we will get only the wanted alpha amino radical. And we could have a substrate scoop in this reaction. We see that it works very well if we have two phenyl groups attached. If we have one phenyl group and one methyl group, it doesn't go as well. If we have two alkyl groups on the amine, not very good yield. If we have one hydrogen and one alkyl group closest to the amine, very bad yield also. And if you look at the scope of electron deficient alkenes, the one that is working best is the diester compound. So here we have also we have compared iron and ruthenium, and we see that if we use the iron photosensitizer, we can do this reaction in 15 hours, 92% using green light. If we do this reaction using the ruthenium BP at its best wavelength, that is actually in the blue region, 450, after 19 hours, we have 64%. So actually, by this means, our iron catalyst, photocatalyst, is actually better than the traditional ruthenium complex. And this took us many years to get to this finding that we actually had a reaction that was better than the traditional noble metal catalyst. And we, of course, did a lot of mechanistic studies. So I will just show you very quickly what we did. We could have quenching of the amines, but we had no quenching of the alkene containing substrate. So in the catalytic cycle, it's only the amine that will be affected by the photoreaction here. So we started from iron 3, we irradiated with green light, we got the excited state of iron 3 that had enough oxidation potential to make the radical cation of the alpha amino TMS compound. we after losing the TMS group, we got the methyl amino radical that makes a Giese reaction. Now we use the ground state again of iron 2 that is actually oxidized itself, but it reduces the radical here to an anion that after workup gives us the product. And we have closed the catalytic cycle here. So if we just make a short conclusion about this, we actually see that we can use a stable TMS methylamine radical and the excited state of our iron-3 complex to do these reactions using a nucleophilic amino radical. And we could also show that we got a higher yield using our iron photosensitizer compared to traditional refining complexes. And just a few minutes that are left, I will talk about another photosensitizers that we have. And that was actually the first one showing photoluminescence of iron back in 2017. And if again, here we have a situation, as you will see here, that we have the iron-3 spectra and we have the iron-2 spectra, and we see at 500 nanometers we actually activate both of them, and that is actually where we are going to use our photoredox box. And we will do something called the atom transfer radical addition. We will add an Rx over a triple or a double bond like this, and we will do this by using light. And one can do this in reductive mode, where we have to have a sacrificial electron donor, or we can do this in oxidative route, and I will route. And if you look at the substrate scope, we see that the best one is when we have a terminal double bond. It gives us this compound in approximately the same yield for both operation modes of this reaction. It goes steric hindrance is not the problem for this double bond. A terminal double bond is attacked before an internal double bond. A triple bond, no problem. And we could actually, in a special case, actually add a bromo compound. And for the mechanism is the following, that we start with an iron-3 complex, and we oxidize that by green light to an iron-2 excited state, and this iron-3 excited state will be reduced to an iron-2 state by triethylamine, and we now get an iron-2 state, and this iron-2 state we will then again oxidize to an iron-2 excited state, and that has enough reduction potential to reduce this Rx compound to an R prime, a radical that makes a Giese reaction, and the product of that actually can act by itself in a propagation chain reaction mechanism here. So we don't need, as you see, we don't need the full equivalent of triethylamine. We have enough radicals now for making a propagation reaction. And in interest of time, I will skip this proof actually that we have in interaction involvement of the two excited state of iron-2 and iron-3 here. Instead, I will just give you the broad picture here, that this is very unique that you involve. You saw in the first photoredox mechanism that we just involved one excited state of the photocatalyst. And here we involve the excited state of both the iron-3 and the iron-2 species. And this is kind of a set mechanism, and this is one of the few examples actually that are in unnatural systems. More common, you see this in photosynthetic system. This set mechanism. So this we think is something very cool, actually, that we use the two excited states here for the photocatalysis. And what we, of course, hope that this could be an efficient method to introduce hydrophobic fluorine groups, because there's no secret that we need this fluorine to get the right redox potentials for our substrates. And also, what is very cool then, that we can use the same irradiation wavelength for both iron 2 and iron 3 LCT states, and we have a non-natural photocatalytic process that has a said scheme. So just some few reflections before I end this, that we have shown also that in iron chemistry, the LMCT state compared to the MLCT state is more useful for photocatalytic applications in general. So this is a change compared to using Nobel system. And we don't need this microsecond lifetime that you saw with ruthenium complexes to get the photocatalysis going. We could use nanoseconds, we could use picoseconds actually for this to happen. And the iron complexes are both good photooxidants and good photoreductants. And then we have the more philosophic question, or a very important question: what is the cost of ruthenium and iridium? I say that this is something bad, but so the advantage is iron, but then we have to set that in relation. What is the cost of synthesizing and making those NHC ligands? And of course, when it comes to photoredox chemistry, the value of the product can motivate the use of ruthenium and iridium. But in other examples that I will not cover today, we have tried to produce hydrogen and so on. If you're going to do that on a large scale, of course, iron could be as more advantageous. And of course, what we're going to now to do is to find new reactions that are catalyzed by our N-heterocyclic carbines. I just want to thank all the people. You saw the people in photos when during the presentation, and many people have been involved, and the funding, and thank you for listening. And if there is time, I can still take some questions. Thanks a lot for listening.
Thanks very much, Kenneth. Great presentation again. Lots of interesting stuff in terms of photoredox catalysis, and to be able to now exploit iron is indeed a worthwhile thing to be doing. But again, I listened to your question, how much is the ligand costing to make at this stage? So you have to look at both parts, that's for sure. So it's open for questions from the audience, either from panelists or from attendees. And again, to remind attendees, you just either need to raise your hand or pop your question into the Q&A. There are already 2 comments in the Q&A, but they're more to do with trying to help Joachim. So I'll get to those when we go back to him. Maybe Joachim, you can have a look at the Q&A, because there's 2 suggestions there as to how you might help solve your computer problem in terms of presenting. And we have a question from Henrik.
Hey, Kenneth, thanks for a lovely presentation. I agree that it's very important to have a look at other options when it comes to replacing the very expensive metals, such as, and iron is a good option here. But I wonder, how do you make these catalysts? You showed one example here where you have this iron boro compound.
That is the tricky part, because iron compared to ruthenium is very much unreactive, reaction with carbene. So when you make ruthenium carbines, for instance, you can use a plethora of organometallic reactions, but here you actually have to make the free carbene. And that is, of course, very sensitive, because a free carbene can dimerize, it can react with itself. So you have to avoid that. And that, if you see in our compounds that we have some methyl groups, and that those methyl groups are there, of course, also to make the carbenes, but they're also there actually to kind of hamper this dimerization. And it's very, very sensitive to the base you use. Basically, the only base that is using it is ultra pure potassium terbutoxide.
Oh,
Long answer to a short question, sorry.
yeah,
But it's complicated.
yeah. We've been also working with the carbons a bit, so we're aware of this Van-Slick equilibrium that can cause troubles. But all right. Thank you for a good answer.
Great. Thanks, Henrik. Any other questions? We're after going a little bit over time there, so we might just pause if that's okay at this stage, Kenneth. And thank you again for your presentation. And we'll move back in the program, if that's all right, to EuChemS. And fingers crossed now that when if you take down your screen there, please, Kenneth, that would be good because we need to share again. If you can unshare.
Yes.
Perfect. Oh, no, not gone yet. So you might have to go to a stop share.
Yeah, I'm trying here.
Yep.
Now.
Yeah, perfect.
Thank you.
EuChemS, we're all rooting for you. Let's hope it works this time.
Indeed. Like so. Is that better?
Hallelujah. Off you go. Good man. Well done.
Thanks a lot. First of all, of course, I would like to thank Henrik and co-workers for allowing me to be here today. I was very happy when I received the invitation, but I have to admit that I was also a little bit surprised because my background is not at all in organic chemistry. In fact, I know very little about organic chemistry. I know a little bit more about how you can control fluorescence in photochromic systems. So in my talk today, you will hear very little about success stories in organic chemistry. You will instead hear a little bit about what you can do when organic chemistry is not working at all. When people hear about photochromic systems or molecular photo switches, they tend to think about something that changes color, and that is of course entirely true. They think about the photochromic lenses, photochromic eyewear that darkens when you go outside and you expose them to UV light. So they do change color. That is the absorption spectra of the molecule changes, but there are also tons of other properties that change when you isomerize the photochromic molecules between the two respective forms. I just listed a set of examples here, and what I'm mostly interested in is how you can control the fluorescence properties of these compounds. This is a very trivial example. In fact, it's referred to as trivial fluorescence switching. We have a fulgimide photo switch that is non-fluorescent in its open form, FGO. It absorbs mainly in the UV region. If you expose this isomer to UV light, it will isomerize to the closed isomer indicated in green. The absorption spectrum redshifts to the visible region. And the closed form FGC, it also emits fluorescence, but with a very, very poor fluorescence quantum yield, 1% only. But in fact, just by using this very simple scheme, you could toggle the fluorescence of the fulgimide on and off just by alternating UV and visible light exposure. Back in 2011, this was sort of state of the art for several reasons, which I will come back to a little bit later in my talk. If you want to increase the fluorescence quantum yield of your constructs, then you could instead rely on FRET reaction, first the resonant energy transfer reactions. Here, what you do is that you typically covalently link a fluorophore to your photo switch. And in one of the isomeric form, there is no overlap, no spectral overlap between the fluorescence of your fluorophore and the absorption of the photo switch. So that's why there is no FRET quenching, and the fluorophore is emitting photons in terms of fluorescence. If you excite it to light at a specific wavelength, you isomerize the photo switch to the isomeric form that absorbs in the visible region, and you have an overlap with the emission of the fluorophore and the absorption of the photo switch, and you turn on the FRET reaction. And as soon as you turn on the FRET reaction, there is no photons emitted by the fluorophore. So this is why you can toggle on and off the fluorescence of your fluorophore that is covalently linked to your photo switch. This is the very first examples along these lines that I did 20 years ago or so during my postdoc period. It is a dihydroindolysine photo switch, which works exactly along these lines. We used a free-base porphyrin fluorophore, and we covalently attached it to the DHI dihydroindolysine photo switch, and it works exactly as anticipated from the cartoon-like sketch on the previous slide. What's interesting is that almost 20 years later, we are pretty much doing the same thing. We are controlling energy transfer reactions by attaching a photo switch to something that is luminescent. The only difference is that 2 years ago we used nanocrystals instead to have a luminescent donor luminophore, and we, instead of FRET, we relied on triplet energy transfer reactions. But the function is very, very much like what we did almost 20 years ago. With these examples that I've showed you so far, we can only change the fluorescence in an on and off fashion. We cannot change the color of the emission at all. Because if we want to change the color of the emission, this scheme is totally useless because we only have one emissive center, in this case, the fluorophore. So what we have to do instead is to use a photo switch that is fluorescent in the FRET active form. So this implies that in one of the isomeric forms, there should be no FRET from the fluorophore to the photo switch. That's why the emission is originating from the fluorophore. Once you isomerize the photo switch to the FRET active form, when FRET kicks in, you sensitize the excitation of the photo switch to the excited state, and in turn, you will observe fluorescence from the photo switch, which has another color. So what you see here to the left is this is the emission of one and the same sample. It spans from blue to almost perfect white light to yellow light, and everything depends on the fraction of your molecules that you convert from the blue fluorescent form to the yellow fluorescent form. And what you see here to the right are the corresponding spectral changes. This is the blue emission from the donor, and this is the yellow emission from the acceptor that is growing in at the expense of the blue emission from the donor. This was very tricky to realize until around 2011, because until 2011 I would say that there were not a single example of photo switches that displayed a substantial fluorescence quantum yield in one of the isomeric form. But Masahiro Iri in 2011 presented this whole beautiful series of DAE, diaryl ethylene derivatives that display fluorescence quantum yields from 22% all the way up to 87%. So what we tried to do here initially was that we wanted to covalently link our fluorophore. In this case, we opted for perilene. For synthetic keys, we decided to make it symmetric. And the whole idea was that when we isomerized the photo switch from the colorless form to the colored form, we would turn on FRET and we would see emission coming out from the DAE photo switch. And this failed miserably due to competing photo-induced electron transfer reaction. We have a little bit of FRET, probably, but very, very little. So instead of having FRET quenching the fluorescence, we had photo-induced electron transfer quenching. And you heard from the talks by Kenneth and Esther before that photo-induced electron transfer, sometimes it's a friend, sometimes it's a foe. In this case, it's definitely a big, big, big foe. So I don't know how many years we spent on trying to introduce novel or other linkers here to screen out or to kill photo-induced electron transfer by distance. We tried adamantyl linkers and bicyclooctyl linkers, but none of these options worked out from a synthetic point of view. So that's why we decided to offer something that we call the cocktail approach. In the cocktail approach, you don't have to covalently link your fluorophore to your photo switch, but instead you encapsulate both these units into polymer micelles. And if you choose your concentration of these two species correctly inside the micelles, the distance between the donors and the acceptors will be small enough for FRET to be efficient, but long enough for photo-induced electron transfer not to occur. So this is an example that I showed you before of the cocktail approach. The more we isomerize of the photo switch, the more we kill of the perylene of the DAE based of the DPA based fluorescence, and we can see a simultaneous increase in the yellow emission from the closed form of the photo switch. If we map these emission spectra in the CIE diagram, we see that we are indeed moving on a straight line from pure blue DPA centered emission to almost pure yellow emission from the photo switch. And on the way there, we pass the coordinates 0.32, 0.33, which is very, very close to perfect white light, which has the coordinate 0.33, 0.33. So encouraged by these findings, we started to think about what if we could realize a full color red, green, blue, or RGB system. What would that require? Well, it would require that we have a blue light emitting fluorophore. And we need to introduce, in addition to the blue emitting fluorophore, a green emitting photo switch together with a red emitting photo switch. And by selectively isomerizing the green emitting photo switch, we could move along this line in the CIE diagram. But if we instead isomerize selectively the red emissive photo switch, we will instead move along this line. And by isomerizing a little bit of both, we were hoping to achieve all available emission color within this triangle in the CIE diagram. What we did was that we opted for a perylene blue emitter in this case, and a green emitting DAE derivative together with a red emitting DAE derivative. And we did the cocktail approach again. We realized that if we exposed the whole tri-component sample to light at 242 nanometer, we could very selectively isomerize the green emitting photo switch without touching the red emitting photo switch. On the contrary, if we instead used light at 415 nanometer for isomerization purposes, we achieved selective isomerization to the red emissive center corresponding to the red emitting photo switch. So this is just a simplified scheme. We start out with blue emission. If we expose the sample to 242 nanometer light, we will isomerize the green emitting photo switch, and we will go from blue fluorescence to green fluorescence. And if we instead use 415 nanometer light, we will go from blue fluorescence to red fluorescence. And long story short, we showed that we could generate all these emission color. And I would like to point out that this emission color comes from one and the same sample using one and the same excitation wavelength for emission readout. So we have a fairly good full color RGB system. The last thing I will tell you about is, we also did this I almost forgot, this is in collaboration with Henrik Sundén. We decided why don't we try to incorporate this system into organogels. That's exactly what we did. And what you see here is that we took a graded neutral density filter and irradiated our gels with. So the blue emission stems from regions where there was very, very little or no UV light used to isomerize the photo switches. And you can see this gradient in the emission color in all three gels. Last thing I will tell you about is an ongoing project. It's about multiphoton microscopy and how we could use FRET and photoisomerization to increase the resolution in multiphoton microscopy. In multiphoton microscopy, it's known that the fluorescence intensity, IF, is not linear to the excitation intensity as it is in the one photon excitation process. But instead, if you use two photon excitation, then it's known that the fluorescence intensity goes as the square of the excitation intensity. If you use three photon excitation intensity, the fluorescence intensity is proportional to the cube of the excitation intensity. In four photon excitation, the fluorescence intensity is proportional it displays a quartic dependence to the excitation intensity. And if you also look at this schematic of the excitation cone used in multiphoton microscopy, it is seen that the intensity of the excitation light, of the laser light, decreases as 1 over c squared, where c is the distance from the focal point or from the focal plane. So this is why when you use two photon excitation, the fluorescence intensity drops as 1 over c4 from the focal point. Three photon excitation, it drops as 1 over c6, and four photon excitation as 1 over c8. So from here, it's clearly seen that the more photons involved in the excitation event, the tighter you localize your emission around the focal point, which implies that the resolution increases with increasing numbers of photons. So this is why 4-photon microscopy has a superior resolution compared to 2-photon microscopy. But 4-photon microscopy is not very common due to the tremendously high laser power required to do so. So we thought that what if we could deliver a technology where we could use 2-photon microscopy but still achieve a resolution that is equivalent to 4-photon microscopy. And this, we refer to this scheme as 4-4-2, 4 for the price of 2. So how do we do this? Because we cannot fool the laws of physics. Well, the whole idea is that we should design and synthesize molecules such that the concentration of the fluorescent molecule also depends quadratically on the excitation light intensity. So in a sense, we sort of merge two individual 2-photon-like processes, and we hope to achieve an overall 4-photon-like response. How do we do this? Well, we simply connect a 2-photon absorbing probe, 2PAP, with a photo switch. This photo switch is FRET active in the thermally stable isomer. So when we start to irradiate the 2-photon absorbing probe with light, there will be no photons at all coming out in terms of fluorescence. But this FRET mechanism don't only kill the fluorescence from your fluorophore. It will also, of course, sensitize the isomerization of the photo switch to the colorless form. This colorless form is not FRET active, so therefore, in this isomeric form, the fluorophore or the 2-photon absorbing probes emit light. So the harder we shoot, the higher the intensity of the excitation light, the higher the concentration of the fluorescent form of the molecule will be. This is why we hope to achieve systems where both the concentration of the fluorescent isomer and the fluorescence intensity of each and every fluorescent molecule displays a quadratic dependence on the excitation intensity. We have quite a few molecules already that works perfectly fine. What you typically do is that you are varying the excitation intensity and you are monitoring the fluorescence intensity. If you do a log-log plot, that is, you plot the logarithm of the fluorescence intensity versus the excitation intensity, and you look at the slope tells you how many photons are there involved in the excitation process. We use 2-photon excitation at 800 nanometers, but we clearly observe a slope that is almost exactly 4, which tells us that we have a 4-photon-like excitation process. This is an ongoing EU project coordinated by Chalmers, Morten Grötli at Gothenburg University is there, a Spanish group led by Uwe Pichel, and then in Belgium we have Johan Hofkens representing the microscopy expertise. So final slide, a lot of people from different universities. I'm not going to explicitly mention any one of them. For money, it's the European Innovation Council, EIC, Swedish Research Council, VR, ERC, and the Carl Tryggers Stiftelse. Of course, thank you all for your kind attention.
Great, Joachim. Thank you very much for that. And again, the presentation is open from questions, either from panelists or from attendees. And again, I remind people, attendees, you can either raise your hand or pop your question into the Q&A box. Doesn't seem to be any questions coming through from either side. Oh, one from Henrik. Over to you, Henrik.
Thank you, Joachim, for an extremely good presentation. I'm curious because we were looking into systems where you could do writing with, high resolution writing. But that was always hampered by that we had diffusion within our samples. And did you ever solve that problem?
Well, what we are typically doing here is that to avoid diffusion, we are using very, very small sample volumes, typically capillary, just to suck up your solution, and then we are looking at extremely small or very small sample volume. So in this case, we can avoid the effect of diffusion, because diffusion will of course distort diffusion will affect your fluorescence intensities in these experiments.
Okay, thank you very much.
Great, thanks, Henrik. So an interesting style of presentation, to have no success stories. I think you're hard on yourself. I think you have many success stories. So well done. Okay, so with that, I'll pass over again to Henrik, and we're going to move on to the penultimate speaker, Daniel Strand. So again, over to you, Henrik.
Okay, thank you, Pat, for giving me the word. The next speaker is going to be Daniel Strand. He's from Lund University, Department of Chemistry. He's gonna tell us about total synthesis, and the title of his talk is Asymmetric Synthesis of ETP natural product from gluonitrine to collective synthesis of thiosylvatanes. And with that, I would like to give the word to you, Daniel. Are you there?
Great. Thank you very much for the kind introduction. Can we first check the technology? Can you see my screen, and is it
Yes.
an appropriate size?
Yeah, all good. And we can see your laser pointer and we can hear you.
That's great. I think we have everything that we need. So first, let me extend a thanks to both Pat and Henrik for putting this together and for the kind introduction. I think it's a great idea, this type of seminar series. Let's see now. Yes, right. So my research group, the heart of what we do is chemical synthesis, and we use that in various contexts with research questions that interest us: chemical biology, catalysis, and materials. But today I'm going to focus on this area, synthesis of complex molecules. And I thought we could start here with the 2,5-diketopiperazine motif. So this is a structure that you will find abundantly in both natural products and in synthetic drugs. So it tends to show up in biologically active architectures. And it's a heterocycle, but what of course is interesting about it is that it has two sp3 hybridized carbons, right? So that implies that we can make chiral derivatives of it, and that is true for what you see on this screen. A type of derivatization that nature seems really excited about is the dithiodiketopiperazines, where you have a thioaminal motif or two across the diketopiperazine ring. And in the context of total synthesis, these have been popular targets, especially the past 15 years, but dating back to the 1970s with work by Kishi, for instance, on gliotoxin. And it's really a bit of who's who of the field. I won't go into more details than that. But one of the reasons these have attracted so much interest is that they have potent biological activities, including anti-tumor, antifungal, antimalarial, viral, and antibacterial properties. From a synthetic standpoint, I will borrow a few lines from Classics 3, that the ETP system is challenging to synthesize, not only because of its dense array of functionalities, but also its liability towards reductants, oxidants, bases, and strong acids. More interesting for the context of this presentation is the second statement, that substrate control of dithiostereochemistry sometimes is possible in the synthesis of more complex target molecules. And I've always found these structures fascinating. They are a bit alien to me. I think many of the people in the audience knows a number of ways to make a carbon-carbon bond or reduce a ketone. But there are not so many ways, that at least I was aware of before entering the area, of how to make aminothiols stereoselectively. So it's a really interesting stereochemical problem as well as a connectivity problem. And if we think about these structures, there's one that stands out a little bit, and it's hyalodendrin. At least in terms of the stereochemical problem, because all stereochemistry in this structure resides in the ETP motif, the central core. And this is, of course, also the business end of the structure. So if you think about this from a functional standpoint, this is really what you're probably interested in to synthesize and then decorate that to achieve selectivity or whatever properties you're interested in. And when we became interested in this area, there was only a single example of asymmetric synthesis of these type of structures, where all the stereochemistry was in the ETP unit, and that was Fukuyama's synthesis of hyalodendrin. And his approach was to use chiral pool. So starting with chiral material and then sending chirality back and forth 2 times across the ring, he was able to make plus heliodendrin. But de novo asymmetric synthesis from achiral components was an unsolved problem. And we were drawn to this problem because we found these molecules, glynitrin A and glynitrin B. So discovered in 2009, they have a unique nitroindolene motif. So glynitrin A has potent antibacterial and cytotoxic activity. I think even more interesting is glynitrin B, that is capable of inhibiting migratory properties in prostate cancer cells. And this is an area that we have been interested in for quite a long time, to selectively target cells with cancer stem cell-like properties or stem cell-like properties. There was also no synthesis reported of these structures. So it was a really interesting synthetic problem from our perspective. And we thought that these were roughly a total synthesis of a problem size that we could manage. So our starting point looked something like this. The key issue seemed to be absolute zero control of the epi disulfide. What we learned along the way is that the indolene unit is extremely oxidation prone. So that is a key synthetic achievement to manage that, or problem to manage that. We have an electrophilic nitrosubstituent. We have an elimination probe serine side chain. And of course, we have this aniline nitrogen that takes a lot of electron density out of the central unit, and that also accentuates the synthetic challenges. So our plan was, as outlined here, to form the tricyclic core with the CH activation reaction, and then simply find a way to construct the epidisulfide using known chemistry, but look at the problem from an asymmetric synthesis standpoint. In practice, that did not turn out very well. So we were able to get the CH activation chemistry going to make this core unit. But try as we might, we were never able to oxidize these 2 positions. And we really tried it all. We did an exhaustive screening of a number of substrates. And what we found was that any basic conditions on this framework led to indole formation. With a lot of the conditions that we tried, we got elimination of the serine side chain. And we were never able to, with any of these, to get more than 8% double oxidation. So 2 years into the problem, to the project, the number of carbon-sulfur bonds that we had formed was exactly 0. And we started to realize that this wasn't so much of a stereochemistry problem, but as of a connectivity problem. This was a really hard type of molecule to make. And if we were going to make any progress on it, we're going to have to rethink the whole strategy. And the solution that we came up with was to switch from diketopiperazine to triketopiperazines as intermediates. That would allow us to do 2 things. We could construct the carbon-sulfur bonds in a stepwise fashion, which would probably facilitate the asymmetric part of it. And we could use the high acidity of the triketopiperazine motif to develop milder conditions for the construction of the ETP unit. And in practice, that turned out to work surprisingly well. So I won't go into the details of the development of this asymmetric sulfonylation reaction. Suffice to say that the challenge for doing anything asymmetrically on this position is that this side and that side are effectively isosteric. So it's really difficult to differentiate between them. And we screened a lot of catalysts looking into that problem, and none of them were able to give us higher selectivity than what you see here, so 60-40-ish. The solution turned out to be that we put a bit of chirality on the sulfur electrophile, because the combination of chirality on the electrophile and chirality on the catalyst was sufficient to induce or to bias the reaction to give us 90-10 selectivity and good yields, the 80s and 90s. And the final reaction conditions are really efficient. You can run the reaction in air. You don't need dry solvents. It takes 15 minutes. So it's a really effective way to install this carbon-sulfur bond. Mechanistically, you can understand this combination of chiral electrophile and nucleophile by forming an activated complex like this. And what happens with that is that you get a rigidified activated intermediate. So we shut down all the conformational flexibility that we had without this methyl group. And that is what allows us to talk to the substrate and relay enough chiral information to bias the reaction effectively. So with that reaction at hand, before setting out to the glyonitrins, we also looked a little bit into the generality, and we screened 6 different substrates, each selected because they mapped on a different natural product. And the selectivity for some substrate dropped a little bit, but since it's a diastereoselective reaction, it's actually still quite practical because you simply separate the minor diastereomer and off you go in the synthesis with an enantiomerically pure intermediate. So there's a decent generality to the method, and again, it's quite practical. The one limitation that we found was that if we don't have an indolene fused unit, there was no catalysis. We needed stoichiometric amounts of the base, and I will get back to that a little bit later in the presentation. So we then went back to the glionitrin problem, and here we started off with this nitroindolene. Acetylation of the nitrogen gives us this intermediate. And what we found here is that the catalyst is sufficiently basic to induce cyclization of this acyclic intermediate to the tricyclic intermediate, and then on to the sulfonylation. And the yield is actually higher running this as a cascade process than it is running it over 2 steps. The only thing that's left then is to install the serine side chain and close the epidisulfide. And we solved that by adding a Grignard to this position. And you can think about the triketopiperazine as 2 amides with a ketone in the middle. So this is the most electrophilic site. And I think it's noteworthy that between these 3 carbonyls and the nitro group, we can, in reasonable yield, add the methyl group selectively to this position. Elimination of the alcohol to the alkene followed by dihydroxylation gave us the right oxidation state on the second, on the 3 position of the structure. From there, we could install the second sulfur atom. And here we were a bit lucky because it added almost exclusively from the desired top face. We are not certain about why that is. It can probably be figured out, but we haven't been able to do it yet. It has in part to do with bias from this side chain. So if you remove this, you don't see the same selectivity, and that will become important later on. The final hurdle to reaching glionitrin was the creation of the sulfur-sulfur bond, and all the known protocols failed. The structure is simply too fragile, so to be oxidized by Ki3 or iodine, for instance. But we found that this type of disulfide effectively closed the sulfur bridge after deprotection of the 2 PMB groups to give us the natural product. And it was a good day, of course, in the lab when we found it. What was bad about it was that it had the wrong absolute configuration. So we had to go back to the start of the synthesis, do it over again, and make the RR glionitrin. So that we actually had the natural product. We also used the same type of sulfur dance to open the sulfur bridge to get to glionitrin B. Treating it with this disulfide opens the bridge, and in the presence of methyl iodide, you get alkylation of the sulfur atoms to reach both structures. So a couple of learning points from this story. We achieved the first synthesis of these 2 natural products. The synthesis in total is 8 or 9 steps and 15% overall yield. The catalytic oxidative sulfonylation reaction is something that opens for asymmetric construction of ETPs on achiral structures, which was really a problem before this work. To get there, we developed a new chiral sulfur electrophile, and we developed protocols for using disulfide reagents to both close and open the disulfide bridges. which is something that we have found since are really general conditions. So getting there, we started thinking about the chemistry that we had developed in a more general sense. Because like I said, the dithiodiketopiperazine motif is something that you will find prevalently in secondary metabolites. And it would be really interesting to see if the practicality of the synthesis of the glynitrins could be translated to a more, to a more general case, for instance, in library synthesis. And to investigate that, we were drawn to the thiosylvatin class of natural products. So that's about 30 members from 23 different strains of fungi, marine and terrestrial. They have a broad range of biological activities also, and particularly, I think, interesting is these type of ring-opened intermediates. They have activity towards the cholinergic system of zebrafish, and I think it's interesting because it's a more specific type of activity than the sort of cytotoxicity that you see of these strongly electrophilic structures. But again, there was no synthesis reported of this class. Strategically, a couple of challenges could be identified at the outset. So the sulfur bridge on the thiosylvatins is more sterically accessible than, for instance, with the glyanitrins, since we have an open position at carbon 3. There's also a electrophilic prenyl side chain that tends to fall off if you provoke it. And of course, intermediates will be epimerization prone through enolization of this position. So the idea here was to initiate the synthesis with an asymmetric sulfonylation following procedures that we have developed, then clean up the stereochemistry, this position, to generate a gateway intermediate from which we could either reach dithiosylvatin or pursue the broader class of thiosylvatins. And other members we envisioned to reach from thiosylvatin, so both through late-stage derivatizations. And again, the bigger picture was to develop a toolbox for how to manipulate this framework for live or more systematic investigations. In practice, this worked surprisingly well. This first trachetopiperazine could be synthesized by direct alkylation with a bit of optimization. So cesium carbonate is critical to avoid the double alkylation at this position. When we looked into the substrate scope in the glynitrin study, we never really optimized it for the more general case. So what we did in this project was that we went back to the reaction and looked at more general trachetopiperazine intermediate. And what we found was that a simple switch of solvent from dichloromethane, as we had used previously, to acetone gave a slightly higher selectivity, but more importantly, catalysis started. So now we have a catalytic system that works with anything that we have tried it with so far. And that gave us this intermediate on a gram scale in 2 steps. Selective reduction of this position with sodium borohydride, followed by sulfonylation, gave us this intermediate. And as expected, we obtained it as a mixture of stereoisomers with a slight kinetic preference for the trans isomer. What is known about such structures is that if the sulfurs are alkylated, there is effectively no energy difference between a cis and a trans geometry. But if they are free thiols, there is a strong preference for the desired cis geometry. There are 3 different mechanisms that one can use to isomerize these positions. You can form an iminium ion here by having the sulfur leave. You can open the ring, so S double bond carbon, or you can enolize. The problem with the first 2 mechanisms is that we can't scramble this stereocenter just because we want to clean up this one, because that means that we lose the absolute stereochemistry that we painstakingly installed in the second step of the synthesis. But the enolization strategy in the end turned out to work surprisingly well. And what we found with a bit of optimization was that during the deprotection of these two sulfurs, if you just leave the reaction going long enough, you can also cleave this methoxy group, but more importantly, you end up with a diastereoconvergent process that gives this as a single stereoisomer. Mechanistically, this proceeds, we believe, through something like this. So you form a boron enolate reversibly, and you generate this type of intermediate, which has a much lower energy compared to this one. The second problem or challenge here is that we need to install the prenyl side chain. The sulfurs, thiols are more nucleophilic than the oxygen, right? So we can't just alkylate this one. We can end up with the wrong product. If we close the bridge first, then basic alkylation of this phenol doesn't work, because the phenol is going to attack intermolecularly the sulfur bridge and cause a polymerization, and all this product, or all our intermediates will disappear. Luckily, we found this paper from Mitsunobu, where he used Mitsunobu conditions to create sulfur-sulfur bonds, and that actually worked really well. So you activate one of the sulfur atoms, you get a transannular closure of the epidisulfide ring, and these conditions are mild enough that we can also induce the O-prenylation as a single pot, and we get dithiosilvatin in one pot from this intermediate. So there was a good day at work, and with that synthesis concluded, we could also start looking at the further derivatization. So alkylation of the sulfurs gives this natural product, and since we don't have a sulfur bridge, now alkylation works fine. So alkylation followed by palladium catalyzed deprotection gives us bilane A. Direct alkylation with this allyl bromide gives us sylvantin-thiol, and the peptide coupling gives us bilane C. So that's 4 more natural products. For the final part of this work, we were interested in the last of the bilane series. And we actually made the proposed structures. Those are the ones that you will see here, bilane D, E, and F. And they were all proposed to be 3s. And what was evidently clear from the spectroscopic data, so NMR spectroscopy data was indistinguishable synthetic from the reported, but the specific rotation was of different magnitude and of opposite sign. And since the relative stereochemistry can be relatively certain, we could be certain about that assignment because they have characteristic chemical shifts. And this alcohol, the secondary alcohol, was assigned by moisture-acid analysis that seemed solid. The remaining sort of ambiguity seemed to be in the absolute configuration of these two. So we figured that the absolute stereochemistry around the ring should be inverted, and then it would also align with all the other thiosilvatins. So that seemed like a reasonable next thing to try. There was also some uncertainty with bilane B, because here there was no stereochemistry assigned for the C2 prime position. So we had a bit of a structural problem to work with also in the final stages of this endeavor. In practice, this is how we approached the problem. We started with the same conditions that we developed for the glyanitrine case, cleaving the sulfur-sulfur bridge and alkylating it. It worked very well to give us this natural product. This can be equilibrated under basic conditions to the trans homologue, which is also natural. To oxidize the double bond, we employed a variation of sharpless asymmetric dihydroxylation. So the ADMX alpha and beta caused decomposition of the structure. They're just too basic. But if you switch the base from potassium carbonate to sodium hydrogen carbonate, we got a very efficient dihydroxylation. If you run this dihydroxylation under mild conditions, so 0 degrees, you can suppress epimerization at the C6 position. But since we were interested in making each of these natural products, we simply let the reaction heat to room temperature generated both stereoisomers here and then converted them to the bilane E and bilane F respectively. So the total yield is 94% in the first step and 77% yield of the subsequent two steps. And now all the spectroscopic data conform to the reported ones. We also use the same protocol for synthesis of bilane B and D. And if you're observant, one of these isomers is the isomer of bilane, the C2 prime isomer of bilane D. So we have B. So we had good reference material, and we were very happy that Bob Capone, who isolated bilane B 20 years ago, had kept a bit of it, and he could share a little bit of authentic material with us. And with that, through HPLC analysis, we could assign the C2 prime position of bilane B as S as well. So to summarize this part, we achieved the first and the collective asymmetric synthesis of dithiosylvatin and 10 of its natural homologs. New conditions improve stereoselectivity and enable catalytic sulfonylation of trachytopiperazine. So it really increases the practicality of that step. A number of optimized methods for late-stage diversification of the thiodiketopiperazine structures, we believe, opens for a more systematic exploration of this 3D pharmacophore. And I think we're doing okay on time. So to summarize that all, a couple of learning points is that we have solved the problem of asymmetric constructions of ETPs on achiral substrates. And a few methodological developments allows us to forge and rupture epidisulfides on really fragile molecular architectures. The approach that I've presented, it's general and it's fairly practical, at least for total synthesis. We have made total natural products and stereoisomers in 5 to 11 steps, should be not 9. And what I think on a personal note is interesting is that we were able to make glyonitrin B in 8 days, which is even less than fermentation, which takes 14 days for that structure. So there might be hope for chemical synthesis also in this context. To leave you with an outstanding question, an anti-selective sulfonylation of trachytopiperazines remains an outstanding challenge, and that's something that we hope to look into going forward. So with that, I will conclude with a couple of acknowledgements. The hero behind the glyonitrin story is Nils Koning. He did all the work there, except for the computational study, which was made by Anders Sundin. The thiosylvatin work was done or spearheaded by Jean Lu, who's a postdoc in my group, and Robin Ekberg, who is a second-year PhD student in my group. We're grateful to Bob Capone, of course, for the authentic sample of bilane B. A shameless plug, we have an open postdoc position. If you're interested in working with these talented people, shoot me an email, and I will be happy to look at it. Vetenskapsrådet and Wenner-Gren-stiftelserna for funding, and all of Europe for your kind attention. Thank you.
Great, Daniel. Excellent stuff. Really cool asymmetric synthesis going on there, and some tough molecules to begin with, that's for sure. So again, the presentation's open for questions. Philip Kahn has raised his hand. So Philip, over to you.
Thanks, Pat. Daniel, beautiful compounds, and I guess fun but a little bit smelly stuff to do. My question would be regarding the 2-thioindole moiety you are creating during your synthesis, and later you have also similarly looking moieties which are, I would guess, prone for elimination of this thioether. Have you ever seen that in your synthesis, or is that a problem during workup? I mean,
just so I can follow, are you thinking about the glyonitrin intermediates?
Either the glyonitrin or, I mean, also in this case, you have a position which would be like adjacent to the...
So here would be an example,
yes,
for instance, right?
Exactly, yeah.
For some reason, we never see elimination, or at least under the conditions that I'm discussing here, we don't see elimination of the sulfurs. But everything prior to those sulfurs tend to eliminate. So I guess they're not as easy to activate. That specific problem is one that we haven't really encountered, other than when I introduced the CH activation pathway, right? Here we see elimination all the time, so that then this is really prone. But the sulfurs, they don't seem to fall off.
Okay, interesting. Thank you.
Great. Thanks, Philip. If I could ask a question, Daniel, a lot of the work employed this new chiral sulfur electrophile. So how easy was it to access that? Was that accessed from the corresponding alcohol, or how did you access it?
It depends when you ask me. If you ask me now, it's straightforward. You have to make the thiol yourself. It's not commercially available, so that's
Sure.
the most painful part. There's a beautiful Nature Chemistry paper on kinetic resolution of that thiol. And that's the method that we're using, because it's not easy to make that thing asymmetrically. The first approach that we used, we simply started from the acetophenone and then did a Mitsunobu reaction to introduce a sulfur on that. That's a Cori protocol, and when you look at it, it looks like Mitsunobu is a stereospecific SN2 reaction, right?
Yeah,
but it came out with 30% EE. So we were still able to make progress with that strategy, because 4 crystallizations and you get 99% EE. So it worked, but now we use the kinetic resolution strategy, and then we make grams of it. So then the problem has gone away.
And do you mind if I ask who reported that paper you talked about?
I wish I could tell you. It's a bit embarrassing that I can't, but I can send you the reference.
Yeah, if you could, that'd be great. Yeah, super.
Yeah, for sure.
So a lot of these compounds remind me a lot of the antimalarial compounds, where there's an oxygen bridge, as you'd mentioned. Those what are they called? Oh, now I'm having a senior moment. Oh gosh, I'll think about it a second. They're well-known antimalarians. They're
artemisinin.
Artemisinin. That's exactly. I should have started with the alphabet and started with A. Artemisinin. And you've made all those thiosulfatins, glionitrin, and bilanes, and they all contain a sulfur-sulfur bridge. Are there any, and this is again maybe a stupid question, are there any natural products that have a sulfur-oxygen bridge?
I cannot give you a answer I'm comfortable with to that question. I'm sure there must be.
Yeah, no, it might even be an interesting synthetic challenge to make those.
Yeah, we have been thinking about analogs of that nature, sulfur, selenium, oxygen, but we haven't dug deeper into it.
Okay, great. So we've a further question from Eric, and then there's a question coming through in the Q&A. So over to you first, Eric.
All right, thank you. Thanks for a very nice and interesting presentation, Daniel. Really, really nice. I had a comment. I mean, I really like these structures. They look really interesting from a biological perspective. Have you, maybe you said and I missed it, but do you have any plans to test them in or make variants of them and test them in different systems?
Yeah, I mean, well, we are gathering sort of a library of them, and the next step is to round off that library and then get them tested. I prefer to do it that way than to continuously send them off, because it gets difficult to compare biological data collected over time. But yeah, for sure. I mean, that's the original justification, and that's definitely where we're going with it.
And where are you? How are you testing them? I mean, is it this cancer effects?
Yeah, I think the most interesting derivatives, I think, are the ones with that are not epithiols, because I think those are just too toxic. And what we have seen in preliminary screenings is that they don't really have any bioavailability. They simply react with whatever sulfur is present, and then they're dead. But the areas that we're interested in is, of course, suppression of cancer cell migration, which is the ring opening variations, the anticholinergic activity, which is also the same class. So I think those are probably where we're going with it. But antibacterial properties is, of course, something that we will be running because it's there.
Just a suggestion. I mean, you could also donate them to some academic libraries, and they'll be screened by different users and might pop up interesting in something along the line.
Yeah, it's a good idea. And on that note, if anyone is interested in some of the material, I think we have quite a lot of most of them. Just shoot me an email and let's see what we can do with it for sure.
Yeah. All right, thank you. Very good, great work.
Great, thanks, Eric. And I have a question in the Q&A from Frederick Dombrowski. Regarding the free thiols, did you also observe the intermolecular formation of a disulfide bond as a side product?
Oh, where are we? Which thiols?
I'm not sure which. I'm just reading.
Okay. Yeah.
Question.
So I think the best answer that I can give is most of those reactions are fairly efficient. We don't see a lot of byproducts in them. That's not to say that nothing forms, but I don't think we saw any of that.
Okay.
I mean, whenever you have less than 5%, it tends to disappear magically. But yeah, we have not isolated anything of that nature yet.
Okay. Good. So perhaps we'll leave it there. Daniel, thanks again. Super presentation. And if you could take down or unshare your presentation, that would be great. And pass over to Henrik, who's got to introduce our final speaker of the day.
Thank you. Thank you very much, Daniel, for a lovely presentation. And also thank you, Pat, for passing over the mic to me. And the next speaker of today, or the last speaker of today, is Philip Klan, who's going to give us a talk entitled Inspired from Nature's Design, from Bioresponsive Tool Compounds and Tumor-Directed Migrostatics. Philip, are you ready to give us your presentation?
Yes, in principle I am. I was just like unmuted. I hope everybody can hear me.
Yep, we can hear you now, and your screen is working fine, and we can see your laser pointer, so all ready to go.
Okay, perfect. Yeah, thanks a lot, Pat and Henrik, for putting this together. I have to apologize. It's a pretty rude behavior to come in late and not attend the other talks. I was unfortunately occupied with some other obligations. Yeah, I hope you apologize for that. So I would like to talk about two topics from my group. One is maybe a bit more known for me, but the other one I think I haven't talked about yet, and I have seen in the list of the audience that there are people who might be really interested in it. Okay, so in general in my group we're fascinated by natural products and the way nature designs compounds. So we share that somehow with Daniel, for example, and we aim to combine certain techniques in order to discover natural product-derived drugs, bioresponsive molecular tool compounds or prodrugs, as well as extracellular targeted drug conjugates or diagnostics which are targeted. And today I would like to have a look at two topics. One is the development of artificial glucosinolates for imaging purposes, as well as a potential prodrug mechanism. And on the other side, I would love to talk about first steps towards bioresponsive microstatics based on cytohalosides. So let's jump into the first topic and first clarify the question, what is a glucosinolate? And you might have already had something today. Most of you might not have lunch, but glucosinolates are part of the herbivore defense system of plants of the order Brassicalis, like broccoli, radish, sprouts, mustard, and so on. And they are produced and stored in so-called S cells in the tissue of the plant. And these cells are adjacent to so-called myrcene cells containing a thioglucosidase in their vacuoles, which is called myrcinase. And whenever now a damage on the tissue by feeding from this little caterpillar takes place, the two components get mixed and the myrcinase is cleaving the thioglucosidic bond of the glucosinolate, releasing a stoichiometric amount of glucose, as well as a thiohydroxymate O-sulfonate A glycon, which can undergo a thiol loss and rearrangement in order to form and isothiocyanate, and ITC. And this is actually the compound which produces this burn on your tongue when you eat fresh radish, when you chew the tissue, or which makes you cry when you had too much wasabi with your sushi. And beyond this feeding deterrent activity, which is predominant, of course, for the producing plant is also using it, especially volatile ITCs, which are self-released by the plant to attract pollinators. And beyond that, there's a bunch of different interesting biological activities spanning from the inhibition of bacterial conjugation, which is the first step, how bacteria conjugate and start to exchange, for example, resistance genes through horizontal gene transfer. We also see a bunch of relatively selective antimicrobial activities. You know, if you eat broccoli, you do not decompose your whole gut microbiome, but you can see that, some bad bugs get affected relatively selectively. And furthermore, there has been a big issue with nosomosis, a fungal infection for Western honeybee populations. And there it has been found if you put these colonies into rapeseed fields, which is also brassicales plants containing glucosinolates and the corresponding ITCs, these insects do not get infected with this fungi. If we're looking into mammalian cells, we have a bunch of interesting activities which could be best summarized as neuroprotective, cardioprotective, and chemoprotective. The targets have also identified to a certain extent, but it's kind of difficult to really exploit and investigate ITCs as potential drugs, because it's hydrolysis sensitive compounds. They are highly reactive. So you would all agree that this is not really a good drug-like candidate. So if you want to work with such compounds, you would probably need to find some form of a prodrug in order to release it at the site where you want to have the action. And actually, there is a natural product. I just introduced it to you. That's a glucosinolate itself. However, the big issue with that is that we as humans, we do not have thioglucosidases in our enzyme repertoire. So we can try to use the bacteria in our gut microbiome to cleave glucosinolates, but if we want to release it at a different position in the body, we would need to have other enzymes being able to release that. And so in order to make ITC activity more exploitable, and also look into tools to use that electrophile for covalent labeling in certain cases, we came up with a concept of pseudoglucosinolates. And I was telling, I'm somewhat regretting that we named it pseudoglucosinolates, because it gets quite difficult to pronunciate when you have more describing adjectives in front of this word, but you'll see it in a second. So what we did was we substituted this glucose moiety by an autoimmolative para-aminobenzyl thiol unit, which we can mask for its 1,6 elimination reactivity with different groups responsive to enzymes or chemical triggers. So whenever this enzyme or chemical trigger is then present, we should remove the masking group, get to the para-aminobenzyl unit, which undergoes the 1,6 autoimmolation reaction, forming the same thiohydroxymate O-sulfonate aglycon, or pseudo-aglycon, if you want, in that case. And this should undergo the same thiolosine rearrangement in order to form the ITC. And the aim here was to generate a platform technology where we can actually use a bunch of different enzymes in order to release ITCs in different contexts. And in order to prove our concept, we started with potentially nitroreductase responsive pseudoglucosinolates. And now I think you get a sense of what I mean, that it was not a good idea to say pseudoglucosinolate. So looking at this, we started with a synthesis, trying to make, first of all, water-soluble compounds. Lorenz and Claire here were leading characters in that synthesis. Well, we started with this chloropec moiety to ensure final water solubility of the probe. Introduced an azide moiety to have a handle for easy click modification, oxidized the other end and attached it to this handle. And then it's actually the important thing is to have this alcohol, which can be oxidized to the corresponding aldehyde. We can transform it into an oxime, and from the oxime we can form in situ the corresponding chlorooxime with NCS, and then do a coupling with the paranitrobenzoyl in order to form the thiohydroxymate as a precursor, and then install the O-sulfonate in the presence of SO3 pyridine complex to form a first potentially nitroreductase responsive pseudo-glucosinolate. And then we have been able to click here different moieties such as danzylamide or this body pi or even biotin to it in order to generate potentially different probes which could be used for different purposes. And so the next step then was to actually verify that this probe is acting as intended, and Claire and Charity were the leading researchers investigating this. So we have used NSFB, which is a nitroreductase occurring in E. coli. It's also a nitroreductase which is responsible for activation of antimicrobial drugs such as metronidazole, or inactivation of other drugs such as chloramphenicol. So it's a relevant enzyme in a biological context, so to say. And when we take our pseudoglucosinolate and then add the enzyme and the cofactors, we can see that we get within our conversion into the corresponding ITC, some other products we can maybe talk later about. And we also see the formation of the corresponding hydroxylamine. That's not unexpected that we get this compound. And finally, both compounds then over time, the hydroxylamine over time also gets converted into the corresponding ITC. However, if we add ammoniac water, we can convert both compounds into the corresponding thiourea as a stable end product. So the general concept seems to work somehow. We were also able to use this Danzyl labeled compounds. Also, they get converted. Again, we see hydroxylamine and ITC. And again, we can also form the corresponding thiourea as a final product. So the general principle seems to work to a certain extent.
Philip, can I just interrupt for a little bit?
Yes.
On the screen, as we're all looking at it, in the right-hand side, there's a black triangle. Can you see that?
Oh, yeah.
Yeah. Can you get rid of that? Yeah, perfect. Great.
Okay. Sorry, that was my timer.
Okay.
Apologize for that. Thanks. So we then try to get other enzymes in place, and the next we tried was the azo reductase. As also here, there are a bunch of bacterial azo reductases which are responsible to inactivate azobenzene containing antibiotics. So also here we first, of course, had to synthesize an azobenzene containing a thiol moiety, and then we started from this aniline precursor doing diazotation, diazocoupling, quite classical synthesis, and then convert the primary alcohol into the corresponding bromide by Apple reaction, did a substitution with potassium thioacetate, and then cleaved the thioacetate under acidic conditions in order to get to this azobenzene containing thiol moiety. And finally, same chemistry, synthesizing the chlorooxime and then coupling to the chlorooxime, and finally installing the O-sulfonate furnished the potentially azo-reductase responsive pseudoglucosinolate with an azide moiety. And when you then do a click with this body pi bearing a terminal alkyne, you can get to this probe here. And there we have a special feature as due to an occurring fret. The compound is non-fluorescent as the emission wavelengths of the body pi is overlapping with the excitation of the azobenzene moiety. So this is a non-fluorescent compound, and in principle, in the moment we cleave that, we should see a fluorescence turn on. Yeah, we will have a look on that in a minute. So also with that, we then had a look on how can we, is the compound functional? And indeed it is. So if we use azo-air from E. coli, it's an enzyme which is not commercially available. So we had to express this with partners. We can see a very clean, much cleaner conversion compared to the nitroreductase responsive compounds into the ITC. And if we then do the next step, adding some ammoniac water, we can also see the clear conversion into the corresponding thiourea. So also the azo-reductase responsive pseudoglucosinolates seem to work. Indeed, I was talking about the fluorescence turn-on. The fluorescence turn-on is happening within minutes and can be, of course, visualized. So this is a probe we could use to actually track or trace azo-reductase associated enzymatic activity. However, the even more interesting question is, can we do something with the ITC we form there? And the first thing which comes up to one's mind, of course, is to use it for protein labeling. And therefore, we did an SDS page gel experiment where we just added external BSA to this solution. And as you can see, we heavily label the BSA. And we actually even suppress any labeling of the converting enzyme, which we have seen. I can tell you that. But so it seems that also this uses as labeling tool compound is potentially possible with these compounds. And furthermore, it was, I mean, I've drawn here a thiourea modification, so it was kind of interesting also to look at what amino acid residues are reacting with these ITCs, and therefore we teamed up with Stefan Sieber and Stefan Hacker, expert in proteome modification, and Mervik constructed this nitroreductase responsive alkyne probe, which we could then activate and incubate with a lysate of S. aureus in order to form thioureas. And then the labeled proteins can be traced by doing a click with an ISO DTB tag, which allows, after certain workflow, to identify the amino acids and the sites which are modified by this probe. And interestingly, it's a very leucine selective probe. So we have 548 sites of leucine which are labeled. Yeah, there is a more complex story behind that, but in order to keep it short, I will not dig too deep into that now. However, finally, it would also be interesting if we can apply these probes in a more let's say, physiological relevant case, or even in vivo. And therefore, we teamed up with a colleague from our institute, Julia Morot-Lekholm, and her PhD student, Leona Cesar. And we use C. elegans in order to show that we can just conduct this activation process in living C. elegans. And I will guide you through this. So we took our probe with a body pie attached, nitroreductase responsive. And the first thing we did was to incubate it with the worms for 1 hour, then have a washout with buffer, and then set the worms in a new feeding plate. And these worms feed on E. coli. And what you see here, this fluorescence there, this is the autofluorescence of the E. coli in the intestines of the worm. So here we pretty much see no difference to the worms which have not seen the probe. So it seems that there's no activation taking place. If we provide an additional phase where we add actually external cofactors, it seems that there is something happening. So what we see here, very slightly labeled, and that was also only the case in a few individuals of the worms, is that we see a very slight labeling of the intestinal membrane of these worms. And it is also reasonable if you consider that the intestines of the worms are full with lysated E. coli, and the external enzyme we actually use to activate our probes is of course present in small amount when you lysate the E. coli, which is the producer of this enzyme. So this result is reasonable. And finally, when we then add our external nitroreductase, we can see clear labeling of this intestinal membrane, and this was visible for all the worms in the test population. And so this labeling is also persistent over days. And interestingly, the ITCs formed do not affect the viability of the worms, neither the healthiness of the daughter generation, which was also kind of an interesting experiment. Okay, so I hope I have convinced you that pseudoglucosinolates might be interesting tool compounds for imaging purposes. However, our current focus on that is that we look into covalent inhibitors being activated by certain clinical relevant enzymes. And with that, I would like to change gears a bit and look with you at our efforts to make first steps towards bioresponsive microstatics based on cytohalazines. And our involvement into this project is based on our participation into the Cytolabs research unit in Germany, where a consortium of 8 different groups from foremostly German universities and the Helmholtz Center for Infection Research are dedicated to provide a systematic investigation and exploitation of cytohalazines. And within this consortium, we are dealing with a total synthesis of 13 cytohalazines, as well as the development of semi-synthetic methodology to modify cytohalazines and design molecular tool compounds for medical purposes, so to say. So cytohalazines are a huge, very diverse class, structurally diverse class of secondary metabolites from fungi, mostly found in genera across the Ascomycota, with the largest portion in Diaphorthe and Chaetomium. However, they're produced by an PKS NRPS synthetase complex, and this leads to a fusion of a polyketide chain with an amino acid derived side chain, and the usual amino acids incorporated are phenylalanine, depicted here in red, or tryptophan in blue, or tyrosine, valine, or alanine. And beyond that, the core structure of these natural products is a tricyclic core consisting out of an isoindolone fragment fused to a macrocycle, which can vary between 14 to actually down to 7 atoms, and these different macrocycle classes are spanning the different subclasses of the cytohalazines according to the nomenclature introduced by Binder et al. Already in 1973. Beyond that, additional structural diversity is introduced to this compound class by different oxidation patterns in the 6-membered ring of the isoindolone core, and here I have depicted a few examples, and of course also additional oxidation patterns in the macrocycle really give you a very diverse and huge class of natural products. The biological activities are similarly interesting and diverse. So we see anti-proliferative activity, anti-angiogenic activity, we see antiviral activity among certain representatives of the class. However, an activity we can find spread over different subclasses and different members of this huge family, reoccurringly is the binding of F-actin and the disruption of F-actin networks. Actin, as microtubuli, are part of the cytoskeleton, and here you can see a cell picture which is stained with phalloidine in red in order to show the F-actin network, and with a fluorescent antibody in green to show the microtubuli as part of the cytoskeleton. While the microtubuli are responsible for, of course, forming the spindle apparatus during mitosis, as well as maintaining an inner cell shape, the actin cytoskeleton part is responsible to maintain an outer cell shape, as well as is heavily involved in the motility of cells, so movement of cells, and also other processes like endocytosis, like trafficking of certain vesicles, and in general, migration of cells. And this is done by a dynamic polymerization of G-actin monomers, which are activated by ATP and then assembled on the barbed end of such an actin filament. So this is a growing end, and then there's a pointed end where these filaments collapse and are disassembled into G-actin monomers. And cytochalassins are believed to bind the barbed end of the actin filament and thereby show their activity. However, the overall structural activity relationship and the picture we have is relatively limited. So, for example, for cytochalazin B, one of the most used compounds in cell biology regarding cytochalassins, direct mode of binding on actin is or has not been known so far. So the closest approximation of this can be derived from a co-crystal structure which was introduced in 2008 by Nair et al., who have co-crystallized cytochalazin D and 11 cytochalazin and they co-crystallized it with a non-polymerizable mutant of G-actin. And this is a co-crystal structure they have found. And what you can see is that there is the N2 nitrogen of the cytochalazin is deeply penetrating the binding pocket. Also, the isoindolone core here is involved in a hydrogen bridge network with a crystal water here. And furthermore, also the C18 OH group as well as the C1 carbonyl group are involved in a hydrogen bridge network with alanine 170 in this non-polymerizable G-actin. So starting in the lack of a better starting point, we started to have a look into how cytochalazin B would fit into that binding pocket. So we did in silico docking, and the essence of it is we found a docking pose where you can actually see that the overall orientation of the cytochalazin B, which is a 14 cytochalasane, so it has a bigger ring here in the macrocycle, is very similar. So also the nitrogen is deeply penetrating. However, the bigger ring size here is not allowing the molecule to penetrate as deep as before. So when we overlay the structures, we can see that the isoindolone core is actually lifted out of the binding pocket, depending on the atom we are looking at, by 1.4 to 1.8 angstrom. And this leads to the fact that some of the hydrogen bridges which we see for cytochalazin D are actually not possible anymore because the distance is just too big. And this leads, of course, to a loss of an affinity, which is also in line with the biological activity on actin between cytochalazin D, which is much more active compared to cytochalazin B. However, with this hypothesis, the N2 NH function as well as the C7 hydroxy function should be very essential for actin binding, while the C20 hydroxy function should not be very essential for binding. And in order to prove that, we did semisynthetic derivatization of cytochalasin B, which we obtained within the consortium through the group of Mark Stadler, who's able to produce these compounds in gram quantities. And starting with that, we have synthesized 4 different derivatives where we have simply alkylated or acylated these positions. So we have made the CB1 derivative with a C7 acetate, which can either be reached by double acetylation in the presence of an excess of acylation reagent, and then we can selectively cleave the more labile C20 acetate, or we can selectively protect the C20 OH functionality with a silyl group, then do the acylation, and finally remove the silyl group in the presence of HF and TBUF in order not to cleave this ester under basic conditions again. Furthermore, we can also selectively acylate the C20 position directly with a derivative CB4. Therefore, we just use stoichiometric amounts of the acylation reagent. And finally, we can address the N2 position by simply deprotonating with sodium hydride and then either doing a methylation reaction leading to CB2 with a N-methylated 2 position, or we can first protect the OTBS, the C20 position as a silyl group, as well as a C7 position, so double silylation, and then introduce an acetyl group to the N2 position, and afterwards cleaving the silyl groups in order to get to CB3. And with these compounds in hand, we then wanted to validate our hypothesis. And what you can see here is that the cytotoxicity, which is not completely related to actin binding, but also is influenced by other activities of cytochalasin B, such as the inhibition of glucose transporters, already gives a hint that our hypothesis seems to be very valid. So we see a little bit of a drop in overall cytotoxicity for CB4, but a huge drop for CB2, which is N-alkylated, and as well as CB1, we see an increase in the value, so a reduced cytotoxicity, which was pointing towards the fact that indeed C7OH and N2NH are positions important for actin binding, while the C20 position is not. However, as I said, cytotoxicity is not the best indicator, so we also had to look into the actual effects on the F-actin network in human osteosarcoma cells. And what you can see here is CB at a low dose, which is defined as the 1 time the IC50, as well as a high dose, which is defined in our system as 5 times the IC50. And what you can see is that it starts that the outer cell shape, the lamellopodia here, collapses, and we see a complete disruption at the high dose. We see a very similar picture for CB4. So the modification in the C20 position doesn't seem to affect the overall activity on actin very much, which is very much in line with our docking hypothesis. We don't see really a big effect on the cells at low dose for CB1, for CB2 or CB3. We see a slight effect for CB2 and CB3 at higher doses, but all of that quite consistent with our overall structure activity hypothesis derived from the docking experiment. Furthermore, we also had a look into the actin polymerization rate, and we did that with a standard pyrene assay where we have a look at the fluorescence over time, increasing when G-actin pyrene conjugated is assembled to the filamentous actin, and then we see pyrene hexamers, which are increasing the fluorescence intensity. And what you can see here is that CB4 and CB are having a very similar behavior. So the acylation in the C20 position doesn't seem to affect the actin polymerization rate very much. Also in the absolute values, you can see that that is very similar. We see some influence for CB3 and 2, which is the N-alkylated compounds, but the CB1 compound didn't even show any influence on the actin polymerization. So with that in hand, we see our general hypothesis confirmed. C7OH as well as N2NH seem to be very crucial for binding, and the C20OH is not really involved and can actually be modified. So what to do now with this structural information? And I would like to introduce you to the concept of microstatics, which has been introduced to the field by Perlikova et al. In 2017, which is pretty much pointing to the fact that hematologic malignancies, considering blood, bone, and lymphatic cancer, are generally characterized by an uncontrolled proliferation, while solid tumors have the additional factor of the formation of metastasis and the invasion of tissue, so invasiveness of solid tumors, so to say. And it's believed, and it has been shown that this invasiveness accounts for around 90% of solid tumor-associated mortality. Contradictory, most of the anti-cancer agent of like the conventional cytotoxic anti-cancer agents are actually targeting proliferation and not invasiveness of tumors. So microstatics are defined as compounds which are actually targeting all pathways which contribute to invasiveness of cancer cells. And there is a belief that, and that has actually been shown, if you combine cytotoxic and microstatic therapy in a complementary way, that this can actually have synergistic effects in cancer treatment, and this holds potential to actually induce a paradigm change in the way how solid tumors are treated, when you not only try to kill the tumor, but you also try to avoid that the tumor spreads in all the body parts, and then finally still leads to relapse and death of the patient. And of course, cytohalazines are interesting in that regard, as they not only target actin disruption and thereby a direct way of cell mobility, but they also have this GLUT1 inhibition, which is an inhibition of glucose transporters, which means that they impair glycolysis in cancer cells, which of course have a high metabolism and very much depending on sugar intake in order to keep their function going. So this is a very interesting dual mode of action. However, you might also know that cell mobility is a very important factor, for example, also for immune cells, in order to keep our immune responses functional and going. So when you apply cytohalazine directly in murine-based anti-cancer therapies, what you can see is that the mice get immunosuppressed. So you need to find a way. So key here is probably to find a way either to deconnect cytotoxicity from microstatic activity, or to make the drugs more selective in order to direct the activity towards the tumor cells and leave out the healthy cells. And so in that sense, we thought about our results and thought, as we have seen the reduction in cytotoxicity for CB1, as well as the complete inactivity regarding actin disruption and the not showing an effect on the actin polymerization, if we now introduce bigger, more stable esters in these positions, such as this benzoquinone TML ester, which is a moiety which is responsive to an enzyme called NQO1. And NQO1 is an oxidoreductase, which is correlated with invasiveness of cancer, and also very, very bad prognosis when expressed in cancer cells. Then we can think of a prodrug being inactive towards healthy cells and being activated by the tumor in order to release cytochalasin B as a microstatic agent at the place where we want to have it. So looking into synthesis, relatively straightforward. Chemistry has been developed in the earlier project. We just silylate the C20OH position. We acylate the C7 position. We cleave the silyl group out, and then we have a BQ-TML-CB in hand. And with this compound in hand, we then first checked if the in vitro activation with NQ1 is taking place. And indeed, within the first 10 minutes, we get an enzyme and cofactor dependent activation into cytochalasin B and the corresponding hydrobenzoquinone core. So that worked out nicely. However, how is it in cells? And therefore, we used NQ1 positive cell lines as well as negative cell lines. And you can see in dependency of the level of the NQ1 expression in the cell line, we see a response to the drug in the proliferation rate. So we are not looking at anti-microstatic activity. We are looking at anti-proliferative activity, which is connected to a certain extent, of course. But what we see is that the NQ1 positive cell lines respond to the compound, while the NQ1 negative cell line does not respond to the compound and shows a very healthy phenotype. So based on that, we also had a look into the incellular effect of these compounds on these cell lines. And what we can see is that the positive cell lines do show an impairment of the F-actin network with structures like lamellopodia already breaking together. And there are different cell lines reacting in different way. We also see multinucleation in these cells, which is a clear sign that the cytoskeleton is heavily affected in these cells. And furthermore, if we look at the NQ1 negative cell line, we can see that there's almost no difference to the DMSO control, while the cytochalasin B, of course, is inducing the changes. So we see we have a very selective prodrug here. We have a NQ1 responsive release. And the next steps here, of course, is to show that it's actually inhibiting cell migration. So we are currently on this to show that. And the future directions of this project will be to increase selectivity by targeting these compounds extracellularly and hopefully exclusively to the cancer cells. And with that, I'm at the end of my talk. I want to thank the most important people. So here you can see the people in the lab. I showed you their participation on the slide. I'm thankful to everybody who has given me money to play on these topics. Thanks to the collaboration partners I have introduced to you on the slides. Thanks again to Pat and Henrik for giving me the opportunity to talk about my research. And I think I'm a little bit over time. Thanks to you for your kind attention.
Well done, Philip. Excellent way to finish off what has been a super day of organic chemistry in Sweden. And again, questions from the audience. I noticed in the Q&A we already have two questions. One from Pavia Herlikova. Apologies if I've got that pronunciation wrong. How do you prevent N-acylation in your selective acylation reactions? In our hands, we usually see both acylation on both nitrogen and oxygen.
Oh, that's interesting to hear. So we don't have experienced that. So when we no, we haven't experienced that. That's interesting. But what we have experienced indeed is when we have bought cytochalasin B from commercial producers, that this compound had different reactivity. And we also had the situation that when the purification of the cytochalasin B after fermentation was leading, or was not complete, and we had some like fatty acids inside, that certain reactions tend to not take place. So reoccurringly, we had problems with impurities, hardly visible impurities, which have led to certain side reactions. However, with the compound we obtained from Mark Stadler, we haven't had any issues in that regard. It's very interesting that you mentioned that.
Okay, and then a second question from Lehos Kovacs. Aminoquinomethane is a rather nasty and elusive substance, and as a reference to a Czech Chem Commune paper then. And the question is, did you have any problem with reductive treatment of 4-aminobenzyl derivatives?
Can you repeat that? The last part?
Yeah. So did you have any problem with reductive treatment of 4-aminobenzyl derivatives?
Reductive treatment of 4-aminobenzyl.
I mean,
maybe I can jump to a slide to.
The questions in the Q&A box. You can have a look at it yourself if you're having trouble catching what I'm saying.
I would say I process it still.
No, that's fine. And the problem with questions when they're just in a box, never quite sure which slide it's related to.
So maybe I mean, I think the question is referring to this para-aminobenzyl unit we have, and then the emulation of that, which is indeed, it's an electrophile again, and I have been questioned with that a lot, because people tend to say this is cytotoxic and you can't use that and so on. I would refer to I think 5 approved antibody drug conjugates containing a citrulline valine PABA linker in their structure, which are all approved drugs for treatment of extracellular targeted cancer therapy. And even if you just take the citrulline valine PABA linker with any payload, doesn't need to be a cytotoxic payload. If you put that on cells, it's not cytotoxic. So yes, I agree. This is a nasty compound when you look at it, but it doesn't seem to affect cells in the way you would think. So we haven't seen any general problems with this compound. I maybe can refer to the fact that that we do not fully understand what these peaks are. We can see masses in there from the cofactors with the ITC, but it might also be reactions in the high concentration here in vitro of this cation with the cofactors. However, in general, I would probably need to deny and say we don't have any bigger problems with that. Yeah. I hope that answers the question somehow.
Yeah, that sounds fine. So maybe, Philip, we'll leave it at that. So if you could unshare your screen, I will just wrap up. So on behalf of the Division of Organic Chemistry, I would like to thank my colleague Henrik for organising today's lineup of speakers. And it's a really nice reflection at this moment in time of both the rising stars and established people in the system in Sweden in organic chemistry. So thanks for organising the speakers, and thanks to all the speakers for indeed giving such outstanding presentations. Our first presentation that we heard was a sort of overview of the importance of organic chemistry in Sweden. So it was interesting to hear about Sheila doing the first esterification. I'll tell my students in first year who are doing and making aspirin by just that type of methodology. And then we had Berzelius, who I have to say didn't look very happy in that photo, one would have to say. Would that be right, Henrik? And you describe him as the father of chemistry in Sweden. We describe Robert Boyle, of course, who was born in Ireland, as the father of chemistry. So we think that's worldwide. So we'll have a little argument perhaps about that. And then it was interesting to see that Niels Lofgren, who I thought was a guitarist in the E Street Band with Bruce Springsteen, actually with Lindquist developed xylocaine, which we've all probably experienced when we go to the dentist. And that was perhaps a good starting point for Astra and the importance perhaps of the pharma industry in Sweden. So then we went into the talks, first from Belen, who gave some excellent examples of her current research in homogeneous catalysis, CH activation, the ability to introduce the magic methyl group where necessary, but also nitrogen, and then being able to extend that out to small molecule drug conjugates. And then more recently, she was involved in carbon-nitrogen bond forming processes. Our second talk then came from Eric, who looked at the chemical regulation of gene expression via small molecule interactions. We heard a lot about G4 stabilizing molecules, and then the success very recently with G4 molecules coupled to these sort of oligonucleotides. So that was a great talk. Then we had Esther, who gave us some very interesting results on her work on lanthanides in synthetic organic chemistry. No funky photophysics going on there, Esther, but there was certainly a lot of new reactants that were developed for reductions and also for the promotion of carbon-carbon bond formations. Then we had Kenneth, who sort of jumped the queue because of technology things with EuChemS, but he gave us excellent examples of his development of iron as an efficient catalyst for row photoredox catalysis, and how really was all down to changing the ligand. You can't change the metal, but you can change the ligand, and the N-heterocyclic carbenes seem to be the trick there, and lots of excellent examples of that, even in Giza reactions, etc. So that was great. Then we went on to EuChemS, who talked to us about controlling fluorescence in photochromic systems. Although he presented it as if he had no success stories at all. I mean, we all learned a lot about some of the failures, but a lot of the excellent work that was done as well. And to hear about 4 photon microscopy being better than 2, and now it's 4 for 2, which seems a little bit of a cheat, but that's a good way to do it. So well done to you, EuChemS. Then penultimate Daniel gave us some excellent asymmetric synthesis of some hard to make molecules, all those glyonitrins, those hylodendrins, those thiosylvatins, and then the bileins, all containing sulfur at key chiral centers, but a lot of sulfur-sulfur linkages as well. So excellent work there and really careful stuff. And as is often the case in natural product chemistry, the compound you make is the enantiomer of what has been reported in the literature. So that's a real pain when that happens, but you know, that's life. And then finally we finished with Philip, again, more natural products. So it was a real flavor of natural products coming through in the end of the presentations, the importance of the isothiocyanate and how that's useful in chemical biology, some semi-synthetic derivatizations then of calecins, and some lot of detailed in vivo work done on the effect of F-actin networks. So certainly from the Division of Organic Chemistry, it's another excellent addition to the series of highlighting chemistry across Europe. So thank you for that. And I'll leave it over to Henrik, perhaps, to give the very final remarks as the local Swedish representative. Henrik, over to you.
Thank you very much. Thank you very much, Pat. I liked your, I like that you picked up the Nils Lövgren connection. Fantastic guitarist. Definitely a fantastic guitarist. I would definitely like to thank all the speakers one more time. I think that they have done an excellent job. I'm also happy that they have answered all my emails and all of that has to do with organizing this seminar. And I don't want to say so many more things because I'm thinking that we are running out of time, but I'm definitely looking forward to the next meeting. And when is that going to be held, Pat?
Yeah, it's got to be in the final quarter, Friday in November, I think. And from my memory, it's Catalonia that will be featured. And then the first quarter of 2025 will be Switzerland. So again, this meeting this morning has been recorded and it will be put up on our website in the EuChemS website, and it'll be also available on YouTube. So you can send the link when you get it to all of your friends and impress them with your work. Okay, so we'll leave it at that and have a nice rest of the day and have a good weekend. So bye from Dublin. Bye-bye.
Bye-bye. Thanks all presenters.
Bye. Thanks.